I love Envato Elements because it is a one-stop shop for me as a content creator, so I can create videos in a short.
I think it's time, madam. It's 2 o'clock.
Principal ma'am, please respond. Shall we start the program?
Shall
Hello?
we start?
Definitely you start the program, no?
Excuse me, Sarah, madam. Someone has to start
Sir,
recording.
tell me. Sir?
Someone has to start recording now.
Okay, sir. Shahjadi madam, please respond. You please start recording, madam. Shahjadi madam.
Okay, madam.
Shahjadi madam. You please start recording, madam. Okay, thank you, madam.
Madam, I have started, madam. I can do it. I can do it. Yeah.
Okay.
Just
Thank
wait
you,
for
sir.
a minute. Just wait for a minute. Because it will
Okay.
take some time to stabilize. I think we can start now, I guess.
A very warm good afternoon to all the honorable delegates, dignitaries, scientists, intellectual experts, and various participants who include colleagues, students, and researchers who have joined in the national webinar on recent concepts and perspectives in mycology today. I'm Dr. N. Lakshmi, lecturer in charge, Department of English, and academic coordinator, taking the privilege to speak as a representative of our college from Raman Degree College, Ramachandrapuram, for the upstart of this webinar. I feel immense pleasure to welcome you all from various regions, disciplinary and interdisciplinary fields to join in this webinar. We are thankful to our Honorable Chief Patron, Dr. Paula Bhaskar Garu, Commissioner of Collegiate Education, under whose guidance such programs are appreciated to be hoisted. He encourages active participation and conduction of statewide, national, and international programs for the benefit of both staff and students for knowledge improvement and orientation. We are also immensely thankful to our Honorable Patron, Dr. C.H. Krishna Garu, RJD, Commissionerate of Collegiate Education, who is present here, who instills in us the spirit to conduct such programs and is always a delightful presence with his valuable insights and guided information. We are equally thankful to our beloved principal, Dr. K.C. Satyalata Madam Garu, who is the torchbearer and guiding soul who encourages us with her pleasant smile, conviction, and certitude to bringing great changes in our college. We are happy to work under her guidance and mentorship of such a dynamic, vibrant, hardworking principal who actively engages in developing college infrastructure and equipment. We, as a unit, both the teaching and non-teaching staff of the college are dedicated towards improving the functioning. I thank the highly intellectual resource persons who are present here, who have spared their valuable time to participate in this webinar. I welcome Dr. Vele Damodar Shenoy, who is a principal scientist and ardent promoter of mycological research, the potent managing director of an international journal, a passionate traveler with a creative edge, from the CSIR National Institute of Oceanography, Regional Center, Visakhapatnam. We welcome you, sir. I would like to render my thanks to Dr. Rohit Sharma, who is an eminent researcher, an active member of various scientific societies, an outstanding reviewer, a remarkable and illustrious professor from IES University and Center for Biodiversity Exploration and Conservation, Bhopal, Madhya Pradesh. Happy to have you with us, sir. I would also like to thank Dr. A. Srinivas Rao Garu, lecturer in charge, Botany Department, Government College of Arts, Rajamahendravaram, who is a great organizer of national symposiums and workshops, an expert in delivery of talks, and has presented many papers. Thank you, sir, for joining in this webinar. The speakers today have been handpicked to provide us with the most up-to-date and relevant information by the active, progressive, and great expertise in her subject, Dr. P. Sara Madam, in charge of the Department of Botany, who had rendered relentless dedication to organize the conduction of this webinar, and I know she would be very happy if all benefit significantly from the presentations of the resource persons. Speaking about the program to be held today, our college proudly announces the inauguration of the national webinar on recent concepts and perspectives in mycology on the occasion of the World Fungus Day, that is today. It's also the occasion of Gandhi Jayanti, and I wish Gandhi Jayanti to all of you. On behalf of all the staff of the college, I extend a warm welcome to all the admirable participants too, who have joined today to make this program a grand success. I request your participation for the entire duration of the program, along with the few opening words. Lastly, I thank the Divine Presence for providing me an opportunity to be a part of this webinar and solicit His blessings for the successful conduction of this webinar. May I now request you all to join in the prayer for a few minutes to be sung by our Vice Principal and in charge of the Department of Mathematics, Kumari Padmaja Garu. Thank you all.
Thank you, ma'am. Tiyurani nabhushayanti purusham न चन्द्रोज्ज्वला न स्नानं न विलेपनं न कुसुमम् नालङ्कृता मूर्धजा वाण्येका समलङ्करोति पुरुषम् या संस्कृता धार्यते क्षीयन्ते खलु भूषणानि सततम् वाग्भूषणं भूषणम्. Over to Lakshmi now.
Thank you, madam. You have mesmerized us with your holy song and also put forth a step towards the upstart of the webinar. May I now request the dynamic Honorable Principal to give her opening remarks and inaugurate the webinar. Over to our Principal Madam, Dr. Professor K.C. Satyalata Madam.
Thank you, ma'am. Honorable Commissioner, sir, Joint Director Garu, AGO Madam, RJD Garu, distinguished guests, resource persons, conveners, learned colleagues, dear students and participants. I wish you all a pleasant good afternoon and hearty welcome to you all to the National Webinar on Recent Concepts and Perspectives in Mycology in view of World Fungus Day. Let me introduce myself. I am Dr. K.C. Satyalata, belongs to Department of Physics, working as principal of this college since July 2021, and I had done my MPhil in 1988 at Sri Venkateswara University, Tirupati, and PhD in 2002 at Sri Krishnadevaraya University, Anantapur. It's my great privilege to speak before this honorable gathering and also to launch the webinar on research concepts and perspectives in mycology. As this college, before starting the webinar, I have to give brief report history of this college. This college is located in the heart of a small semi-rural town, Ramachandrapuram, situated in Dr. B.R. Ambedkar Konasima district, the rice bowl of India. More than 80% of population are farmers here. The place is surrounded by lush green rice fields and luxuriant vegetation. It is also a bustling commercial center for clusters of surrounding villages. Realizing the need for better educational facilities and opportunities for its rural youth, the legendary person of this town, Sri Kuttivinti Perraza Pantulu, founded educational trust named as Sri KPP Educational Trust on 21/9/1906, and a land of 5.25 acres was allotted to this college by that SKPP trust for the construction of permanent buildings. Actually, this college was established in 2014 with only 4 courses, BSc, Mainstreams, MPC, and BJC, BCom General, and BA HCP with initial student strength of 128 to provide quality of education to rural students. Now, the college is offering 8 academic programs with 580 students catering to the needs of socially and functioning, marginalized and unprivileged sections of the society. The college was created with an objective of providing quality of education through CSP, internship, apprenticeships and trainings and placements with practical orientation as per national new educational policy. The college is working under the able guidance of our Honorable Commissioner, sir, operating with a vision of enabling students to thrive not only in academics but also in character building, leadership, extracurricular activities, and sports. In addition to all, a strong, dedicated, highly qualified, and capable team of faculty are the driving force in the growth of the number of students and also the institution itself. Let me relate the concept of the webinar to the basics of science and its relation to mycology in brief. Knowledge of science and scientific developments about our earth, knowledge of ecosystems and biodiversity, and interaction of humans with ecosystems are important to help us understand how to manage our planet for a peaceful and sustainable future. Sustainability is a basis for human existence, and this concept has potentially made mankind to involve in various new challenges and research which led to the development of new branches in the field of botany and its interdisciplinary fields like biotechnology. Present topic of the webinar is recent concepts and perspectives in mycology. Mycology comes from the Greek word myces, which means fungi, and ology means the study of. That means the study of fungi, a group that includes the mushrooms, yeasts, etc. Henry Anthony de Perry is the father of mycology, and he discovered the life history of fungi and studied their role in disease. Henry was a botanist, mycologist, and also microbiologist. He is also known as father of plant pathology. And the applications of this mycology are many fungi are useful in medicine and industry. Mycological research has led to the development of antibiotic drugs such as penicillin, streptomycin, tetracycline, etc., as well as other drugs including statins. As we know, that is the cholesterol lowering drugs. Speaking about job creation for the students and researchers, mycology has major applications in dairy, wine, baking industries, dyes and inks production, chemical industries, the pharma sector, forensics, etc. Mainly, as we know about mushrooms, which belongs to fungi family, which produce large amounts of vitamin D when exposed to ultraviolet light. Speaking about careers in mycology, it involves number one, academic, research and teaching, number two, conservation and ecology, number three, biotechnology and industry. With this briefing, I would like to conclude my opening remarks about the webinar. I feel honored to welcome all the resource persons once again, the intellectuals, researchers, and colleagues, students for taking a keen part in this important program. I am sure that you all will feel enriched with knowledge after completion of this webinar. I hope you all find this seminar highly engaging, fruitful, and beneficial for your future ventures. Thank you. Thank you, one and all.
Thank you, Principal Madam. It was Dr. K.C. Satyalata Madam providing the inaugural address of the particular webinar and introducing to all the way in which major applications are being carried out in the particular field of mycology and what mycology is and the different sustainability aspects also she has spoken about, along with a briefing out of the college and its features, along with its vision. Thank you, Madam, for carrying on with this inaugural address and opening remarks. Now, I would like to request our honorable, learned intellectual, a worthy, deserving administrator in the form of our RJD of Zone 1 and 2, Dr. Chappadi Krishna Garu, the patron of this webinar, to say a few words to initiate the webinar and present the inaugural address for the webinar. Over to you, sir.
Is it disconnected, ma? Krishna garu?
Yes, ma'am. Yes, ma'am.
I think so. So, one person can ask him to join. I think.
Yes, madam. లేదు. Sir is joining, ma'am.
Hello, madam.
Yes, sir, you are audible. You please continue,
Yes.
sir.
Okay, madam. Good afternoon to everybody. Seminar President Dr. K.C. Sacharata, Principal GDC Ramchandrapuram, and the Seminar Convener Dr. Sala Parpatti, Lecturer in Botany, and IKC Coordinator Dr. N. Srinivas, and Academic Coordinator Dr. N. Lakshmi, and Vice Principal Madam, and other participants and resource persons. Good afternoon to all. Greetings to the participants of the national webinar on recent concepts and perspectives of mycology. We all know mycology, the scientific study of fungi. The kingdom fungi includes an enormous variety of living organisms, collectively referred to as true fungi. Scientists have identified about 1 lakh species of fungi. This is only a fraction of the 1.5 million species of fungus probably present on Earth. Edible mushrooms, yeast, black mold, and the producer of the antibiotic Penicillium notatum, all are members of the kingdom fungi, which belongs to the domain Euchera. Fungi are diverse and ubiquitous microorganisms with critical ecological, medical, agriculture, and industrial importance. Mycologists investigate various aspects of fungi, including their taxonomy, philosophy, physiology, ecology, genetics, and applications. Mycologists engage in public education to promote awareness of fungi and their importance. Citizen science initiatives also contribute to fungal research. In mycology, a multidisciplinary approach is often necessary as fungi are incredibly diverse and have far-reaching ecological and economic importance. Researchers in this field contribute to our understanding of the natural world and develop practical solutions to various challenges. Fungi play diverse and crucial role in benefiting mankind across various aspects of life, including agriculture, medicine, industry, and environmental sustainability. Why fungi are important for humanity? Food production. Edible mushrooms. Many fungi, such as oyster and button mushrooms, are not only nutritious but also delicious. They are a significant source of protein for many people worldwide. In medicine, antibiotics. Penicillin derived from the fungus Penicillium revolutionized medicine by treating bacterial infections. Fungal products like cyclosporin are vital for organ transplant recipients. Some cholesterol lowering drugs are produced using fungi. Fungi are used to produce enzymes like amylases, cellulases, and proteases, which find applications in various industries, including biofuel production, textiles, and detergents. Some fungi are sources of essential nutrients like B vitamin, making them important in dietary supplements. Fungi can help clean up environmental pollution by breaking down and absorbing contaminants from soil and water. They are used in myco remediation to tackle issues like oil spills and toxic waste. Fungi from symbiotic relationship with plant roots enhancing nutrient uptake. This is critical for crop growth. Fungi are employed in composting process of breakdown organic matter, reducing landfill waste. Fungi are integral part of ecosystems and can be indicators of ecosystem health. Conserving fungi is essential for preserving biodiversity. Fungi are incredibly diverse and their potential benefits to humanity are still being explored. However, it is important to note that while fungi offer numerous advantages, some can also be harmful as pathogens, causing diseases in humans, animals, and plants. Understanding and managing fungal interactions with humans and the environment is a critical aspect of mycology and biology in general. I congratulate the principal, Dr. K.C. Sachalata Madam, and convener, Dr. Sala Parpati of the webinar for the choosing novel concept for the webinar, which is the need of the hour. Hope the resource persons across India enlighten the participants with insights of fungal world. All the best to the participants. Over to Lakshmi Madam.
Thank you so much, sir. It was a nice elaboration on the term mycology and its various underlying applications. And also you have elaborated on the role of fungi and the various aspects related to it. It was a very stimulative and encouraging remarks which are made by you. Thank you so much, sir, for joining with all of us in this webinar. Now I would like to request the epitome of intellectuality who has burned the midnight oil to make this webinar a reality. It is none other than Dr. P. Sara Madam to speak about the webinar, to introduce the topic, and also to enlighten both the accustomed and unaccustomed participants of the college and the outside world about this new topic. Over to you, Sara Madam.
Thank you, madam. Good afternoon to respected RJD Dr. C. Krishna Garu, Principal Dr. K.C. Satyalata Garu, respected resource persons Dr. Belle Damodara Shanai sir, Dr. Rohit Sharma sir, Dr. A. Srinivas sir, all the faculty and dear students. This is Sara Palapati working as botany lecturer in Government Degree College, Ramchandrapuram. Today, we are celebrating World Fungus Day. The world is visually dominated by animals and plants, but this world would not have been possible without fungi. Fungi play a crucial role in the balance of ecosystems. They're essential to the recycling of nutrients in all terrestrial habitats because they are the dominant decomposers. Humans use fungi for many purposes, already Principal Madam and RJD sir told. But most of common people and even many biologists do not, however, realize the importance of fungi. So the awareness of fungi about this vital must be spread throughout the world. If people know something about fungi, there is a much stronger chance that they will protect them for the future ecosystem, indeed for betterment of mankind. In this context, British Mycological Society has taken initiative to organize World Fungus Day on 2nd October. The main aim to put an event for introducing fungi to the general public. India is having great legacy of mycologists and must get involved in the motto that conservation of fungi. Hence, the most prestigious government degree college, Ramchandrapuram, come forward to organize national webinar on recent concepts and perspectives in mycology, RCPM 2023. I hope this webinar would help to gain more knowledge about mycology through eminent persons in the field of mycology. I am thankful to MycoAsia Group on behalf of Government Degree College Ramchandrapuram, who have shared their platform through Google Meet for conducting this webinar. Thank you one and all.
Thank you, Madam Saraji, for pointing out to the very intricacies of fungi as decomposers and also underlying their contribution to the balance of the ecosystem and their usage for sustainability. Thank you, Madam, for introducing us to the particular concept of the webinar. Now, I would like to request Shri T. Timothy Garu, lecturer in zoology, who is an expert in zoology and aquasciences, and also as a great advisor of balanced diet in the college, and also is a nice human being, along with his ingenuity in handling students and promoting educational values. Over to you, Timothy, sir.
Thank you, ma'am. Good afternoon to all. This is K. Timothy, working as a lecturer in zoology, Government Degree College, Ramachandrapuram. Is very happy to introduce eminent personality, Dr. B. Damodar Shanai. He is a well-known mycologist and principal scientist working at CSIR, National Institute of Oceanography Regional Center, Visakhapatnam. He is the managing editor of Mycology journal, MycoAsia. He is expert in marine biodiversity, biotechnology, molecular biology, and marine bioinformatics. Also, expert in fungi, having published more than 45 papers in international journals. To his credit, Dr. Bilal Shanai is on the advisory board of Amity Institute of Oceanography and Atmospheric Sciences, India. Dr. Shanai has a keen interest in promoting mycology among children and youth. He is very active on social media such as Facebook, Instagram, Twitter, etc. to promoting mycology and awareness. His non-specific hobbies include traveling and exploring India and writing poems. Now, I request Dr. Damodar Shanai to share his views and ideas about recent concepts and perspectives in mycology. Thank you.
Thank you, Timothy, sir. Thank you, sir, for providing us just about the greatness of Dr. Belle Shanai. And we are very happy to have him with us. I would like to warmly welcome you, sir, as an ardent promoter of mycological research and a passionate traveler with a creative edge, and also having publications and being the managing editor. It costs a lot, and I feel that you have great expertise in the field. So I hope all the participants will take advantage of this opportunity to grow and learn together with the introduction of your topic. Over to you, Shanai, sir.
Thank you. Okay, am I audible?
Audible, sir.
Yeah, thank you. Thank you very much, Dr. Timothy, for introducing me to today's audience. In response to your kind words for me, I would like to cite a Telugu sentence. That would be Endaru Mahanu Bavalu Andariki Vandanam. I mean, I'm happy to be amidst the learned people here. And I'm very much thankful to the management, the team of Government Degree College, Ramachandrapuram, for giving me this wonderful opportunity and to share my ideas on fungi. I want to congratulate the college, Government Degree College, Ramachandrapuram, for celebrating World Fungus Day. As I witness, the World Fungus Day is being celebrated not only by the students and teachers of zoology and botany. We have people from English department. We have people who are in physics department, who are from the physics field. So actually joining us in celebrating World Fungus Day. That is very, very much a very pleasant thing to actually witness. So thank you very much. Once again, I will be loading my presentation slides and I will be talking for next 30-35 minutes using presentation slides. I mean, as I know, this is the bulk of the audience would be bachelor's degree students. So I will be talking. My presentation is titled as Welcome to the World of Fungi. Basically, it means my presentation would be for kind of beginners in mycology, or I would people who may have some very basic information about mycology. But I will be adding up some of the recent advancements in mycology. Thank you very much again. Be patient while I add my load my slides. I hope the first slide is visible. The slide which says "Welcome to the world of fungi." Visible, sir?
Yes, sir.
Visible, sir.
Okay, great. Okay, so the next 30 minutes would be playing with the presentation slides and talking about fungi and other nice stuff. Actually, fine. What is the plan today? My plan would be to first tell who is a mycologist. I mean, I will be talking about the traditional view and also adding up with the recent improved understanding of who is a mycologist. Then I'll move on to the core concept of mycology. What is mycology? How the mycology has changed over time. In brief, actually, I'll be discussing that in brief. Then I'll be talking about what we have been doing at CSIR, National Institute of Oceanography, with respect to fungi. I'll be giving some ideas about the research we have been doing about the fungi associated with a marine pollutant called tar balls. I will tell what are tar balls in one of incoming slides. And at the end of my presentation, maybe after 30 minutes, if you feel inspired, or if you get motivated, or if you find fungi to be interesting, then I will give you some tips how to become a mycologist. Mycologist, as defined in a traditional way, mycologist in a modern, understanding or modern definition. Okay, so lastly, I would be giving some resource. I will be pointing at some online resources that you could look at to improve your understanding, or maybe get up-to-date information about the various things what is happening in the field of mycology. So, who is a mycologist? Very simple question. The person who studies fungi is a mycologist. Okay, but that is the old way of defining a mycologist. The new way includes, it's a very liberal way of including people under this term mycologist. In the new term, in the new definition, actually mycologists, I mean the mycologists are these people. Mycologists are the scientists, researchers, educators who study fungi. So this includes everyone from ordinary citizens who study and explore the world of fungi in their everyday lives, or to lifetime experts like our own Dr. Rohit Sharma. Okay, so basically in a modern day definition, the mycologists include scientists, researchers, educators like Dr. Sarah, and ordinary citizens and lifelong experts who study ordinary citizens and lifelong experts who are devoting their time understanding the life of the kingdom fungi, actually. As I defined, mycology is nothing but the study of fungi. Why? And I would like to highlight here, and other speakers also have highlighted that fungi are underappreciated compared to bacteria, compared to viruses, especially after the recent COVID waves. The viruses, the importance of studying viruses have become very much critical. So comparatively, the fungi are underappreciated. They are the poor cousins of bacteria and viruses, actually. So there are many reasons for this. One of them is there's many of us, actually, many people, many common people, general public are not aware of the kind of contributions fungi do actually in their day-to-day life. The public, that awareness of fungi and their contributions is not that great. This is more so among general public and non-mycologists. Of course, mycologists should have some understanding of it, but like physics people, people from English department, they may not be fully aware what is happening. In terms of mycology, how fungi are important, how fungi contribute, how fungi play important roles in our day-to-day life. So therefore, as Madam Sarah was telling, the British mycologists decided that we should have one day for the fungi. So therefore, October 2nd is celebrated as World Fungus Day, and we people at Mycology have been celebrating for last 2-3 years. And as you know, today is the birthday of Mahatma Gandhi ji. So it's important that we remember the fungi along with Mahatma Gandhi ji on the October 2nd, actually. And many programs have been going on since yesterday to celebrate the World Fungus Day. Even we at Myco India have conducted a series of lectures. There are 12 lectures on different aspects of mycology. If you miss the lectures, you can go to our YouTube channel after a week. Then you could look at the recordings, actually. So coming back to the life of diversity of life on this earth, this is a tree of life. If we have to graphically represent diversity of life present on this earth, you could use this graph on this picture. This is the tree of life. You have a hypothetical ancestor here, and you have animals. You have butterflies. That means basically this is where we human beings belong, somewhere here. And if you see that just below the animals, you have a branch for mushrooms and fungi, basically for fungi. And here is a branch for, plants. Evolutionarily, fungi are closer to animals than to plants. So we might have been studying fungi in the botany department traditionally, but the evolution says, the evolutionary evidence, the recent one says that fungi are closer to animals than to plants. So this would have implications when we actually develop antifungal agents. So this will have some implications in that. So if you ask, to a beginner, my classification of to a non-expert, to a general common person, my definition of my classification of fungi would be like this. Fungi could be classified into 3 groups for practical purposes. First would be mushrooms, which are macroscopic, and they have got the multicellular cell system. Then would be molds. Of course, mushrooms are visible. You find mushrooms almost everywhere, and they are beautiful. They are beautiful, and they are attractive. People love mushrooms, and many people eat mushrooms. So mushrooms are the first thing you can use mushrooms as an example to introduce the non-experts to the field of mycology. Second would be molds. Molds, these are microscopic. You cannot see through your naked eye. So you need microscopes, and you would see that these are multicellular. And you can find molds in our home dust or on our bread. You see the green thing, but the actual structures you could see under the microscope only. The third group would be yeast. These are mostly unicellular, but when they divide, they would be, bicellular. So these are the major groups of fungi, according to me, for when we take mycology to common people. Mushrooms, molds, and yeast. That's great. As I told you, animals and fungi are each other's closest relatives. So that there is a paradigm shift in our understanding of relationship between fungi and animals and plants. Okay. Going back to that older times, when I'm talking about millions of years back, actually, long before the plants came on the land, from the aquatic system, Earth was covered by giant mushrooms. Basically, the ancient world was full of mushrooms. Mushrooms, as I told you, are a kind of fungi. So the ancient world was full of mushrooms. Okay. Even before the plants, big plants came onto the earth. So this is based on fossil data. So what does it mean? That means fungi were one of the earliest inhabitants of this earth. Actually, after that, the big plants, emerged. Emerged in the sense there was a competition between these giant mushrooms and fungi and the plants. So that somehow plants won the battle because they could use the nutrients in a better way. So in today's world, you find trees, more plants probably than mushrooms, or at least the giant mushrooms. That's because somehow the plants have beaten or overtaken the giant mushrooms. Of course, mushrooms are there, but these may not be as giant as probably some meters. And if you didn't know, the largest living organism on this earth is a fungus. So this is not Dr. Shena telling. This is a Guinness Book World Record or a Guinness World Record. The largest living organism on this earth is a fungus. That's a mushroom. The name of the common name of that fungus or mushroom is honey mushroom. The scientific name is Armillaria hosti. Why it? And it was reported from United States. So why is this particular mushroom declared as the largest living organism? As you know, when you see a mushroom body, that is just the visible part of it at the above ground. But the below ground, there would be interconnected mycelia, the mycelial network. In this case, the mycelial network of the honey mushroom would cover a total area of 965 hectares. That is 2,385 acres, and which is equivalent to 1,350 football stadiums. So this is reported from one national forest, Oregon in the USA. Actually, okay. So the mycelial network is around 1 meter underground, and it has been calculated that this particular fungus could be as old as 2,400 years. For last 2,400 years, this fungus has been living on this earth. Though human lifespan is around 78 years, this particular fungus has been living for last at least 2,400 years, and it has been covering an area of almost 1,350 football stadium. Isn't it amazing? And therefore it holds the record of the world's largest living organism. Actually, okay. Now going back to the COVID pandemic, especially in the second wave of COVID pandemic, you could have read about in newspaper about black fungus. See, the phrase black fungus is incorrect, and actually it reflects the poor understanding of mycology among non-experts, and especially in the news reporters and the public, general public. Because in this case, the fungus is hyaline. Okay, the fungus is not black. The fungus is hyaline, but the symptoms caused by that particular fungus or group of fungi are black. Therefore, it is incorrectly known as black fungus. So it's about the name. But what was the importance of this fungus or a group of fungi? Actually, they killed this particular fungus, resulted in mucormycosis. Okay, and that resulted in the death of 4,500 people during the COVID pandemic, because immunocompromised individuals, especially COVID patients, were attacked by this kind of, not physically attacked, infected by this black fungus, incorrectly called black fungus or mucor species, and resulted in death of at least 4,300 people. So basically, I'm trying to tell you that people's awareness about mycology is not that great, and that is because the mycologists are not communicating the important things to non-experts and to general public. We mycologists have to do more work towards the public awareness. Therefore, I am going everywhere. Wherever people call me, I'll go and talk about fungi and their importance. And next one would be, somebody was, a couple of the earlier speakers were mentioning about penicillin. Of course, this is a very beautiful story, for a very motivating story for mycology, because the first antibiotic, the penicillin, was from the fungus, and that's from Penicillium rubens. Somebody was mentioning about Penicillium chrysogenum. I will tell you what has happened to that name actually. So recently, when mycologists did the taxonomic revisions, they did the culturing of that particular fungal culture, which was used by Fleming to produce penicillin. They did the micromorphology studies, and this is how the fungus looks under microscope. They did the sequencing, DNA sequencing. They actually sequenced a gene called beta-tubulin, and they constructed a phylogenetic tree. When you construct a phylogenetic tree, you actually find out the relationships, revolutionary relationships of a group of organisms. In this case, it was found that Fleming's penicillin producing fungal culture actually belongs to the clade of Penicillium rubens. Therefore, the new name of that fungus is Penicillium rubens, not chrysogenum, okay, not notatum. So please remember the recent advancement in mycology based on the molecular phylogenetics. So this would have implications. When students are taught the wrong names, they pick up the wrong information. Therefore, correct name should be communicated when we actually talk about mycology. And this should start at the college level itself. And I think in colleges, we are still teaching them fungal classification as Ascomycota, Basidiomycota, Zygomycota, Chytridiomycota, and Deuteromycota. I think those things are changed. Those things have changed. I'll come to that later. What I'm going to tell here is that penicillin was the one of the molecules from the fungal origin. Actually, there are many more drugs, medicines are discovered from fungal origins. What are those? Here is a list of FDA USA approved medicines from fungi. So, for example, here is one medicine for cholesterol lowering, another for bacterial infection. Third one for terminal illness. I will talk about psilocybin, terminal illness. I'll tell you what does the psilocybin do actually. One more somebody was mentioning about cyclosporin used in during organ transplantation, and that is the market value of that molecule is at $1 billion US. 1 billion. Okay, so there are more of molecules for fungal infections, cancer treatment and prevention, and migraine headaches. So you can use. There have been so many advancement in the drug discovery from fungal sources. So this is just a small list of FDA approved medicines from fungi. About psilocybin, these are derived from, they extracted from a set of mushrooms known as magic mushrooms. Why they are called magic mushrooms? When you consume the extract, actually it gives kind of hallucination. So that makes you feel light. One of the components of that is psilocybin. What does it do? it can help you. It can help in treating terminal illness. There is one compound from this magic mushroom that can be used to treat drug resistant depression. So it is not yet marketed. There is a potential in these kind of mushrooms. Actually, those are known as magic mushrooms. So as many of you know, this is a common sight in our households. Bread unused for some days, you could find this kind of molds, bread molds. Then when while here in this case, in this case actually, the fungus is actually infesting and destroying our food. In second situation, the fungus itself becomes our food, the ME mushrooms. So how about mushrooms? Indians are consuming mushrooms, but not that much. That's because of our certain biases about eating mushrooms. Second thing is that comparatively China is producing almost 70% of the world's mushroom in market for the market, world market. If comparatively India is only producing 2% of the contributing only 2% to the world's mushroom production. Okay, but the US market, the global market, mushroom market is valued. It's a big market. The world mushroom market is very big. It is valued at $46.1 billion US dollars. It was in the year 2020. That's a big amount of money. It's the actual market, that is the value of the market. And this is the global mushroom market is expected to grow at around 9.5%, in next 5 years, from 2023 to 2028. Basically, this value will go up maybe up to 65 billion US dollars. If I'm not wrong in mathematics. Okay, so that's how big is the global mushroom market. So mushroom cultivation should be popularized because the market is pretty good and it is expected to grow at 9.5%. So Indians can get into mushroom cultivation, not only button mushrooms, other sorts of mushrooms also, or maybe shiitake, or maybe magic mushroom, not for the consumption, for medicinal actually. About some scientific information about mushrooms. So we often get this question, the people send us mushroom pictures. Is it edible? I think it's a very dangerous thing to tell people just looking at the images that this particular mushroom is edible or not, because similar looking mushrooms can have different effects on people actually. Therefore we avoid telling it is, especially wild mushrooms. We don't actually tell. We advise people to eat unless and until it is properly investigated by the experts. So here is a list of around 2,000. Here, in this paper, there is a list of 2,100 edible mushroom species. So this is based on the worldwide data. They have reviewed the data. They have listed around 2,100 edible mushroom species. They say these many mushroom species could be safe to eat. Safe to eat. But as you know, we have mycologists have already described around 15,000 mushroom species. Out of them, only 2,100 are confirmed to be edible. So what can we do with the remaining, say 13,000 species of mushrooms? Of course, many of them are known as poisonous mushroom, for example, Amanita. But we can go from 2,100 number to 3,000 if we use our traditional knowledge. See, the edibility of the mushroom more or less is based on the trial and error from the traditional knowledge. Best would be to talk to your grandparents or elder people in the community, and if the community is inclined towards eating mushrooms, that knowledge would be very useful, and documentation should be done actually. That probably our CSR labs or TKDL division is planning something. I was talking about 15,000 mushroom species that we have described. And of course, we have so far described around 2 lakh fungal species. If you take the conservative figure of 15 lakh estimated or guesstimated fungal species that could be present on this earth, still our understanding of fungal diversity is quite poor. Basically, we know around 15 divided by 2 lakhs, right? No, 2 lakhs divided by 15. That would be around 20%. So our understanding is quite poor with respect to fungal diversity. So we know about 2 lakh fungal species, and we know that there could be at least 15 lakh fungal species on the earth. So where are the missing fungi? They could be in a tropical country like India, because there have been lots of studies in Indian Indian habitats, but the exploration can go to a different level, and we can add to the list of missing fungi. Basically, we can discover the missing fungi in Indian habitats. What we have been doing at CSIR NIO, CSIR National Institute of Oceanography, we have been studying fungi present in the seawater or sea sediments, oceans actually. We think so. Oceans are one of the possible areas where we can discover these missing fungi actually. So you can find marine fungi, even though they are evolutionarily polyphyletic. Polyphyletic means they are not belonging to only one family or one genus. They are belonging to so many classes, or but they are loosely called as marine fungi because they are found in marine environment actually. So you can find marine fungi on driftwood, mangrove wood, roots, pneumatophores, seedlings, leaves of mangrove plants, soil, sediments in marine environment, seawater, dead and decomposing animal substrata. We at NIO, National Institute of Oceanography, we go to the open ocean and we collect water and sediments from as deep as 5 kilometers below sea surface. Then we, at that particular depth, the temperature is around 4 degrees Celsius or 2 degrees Celsius. And imagine, as you know, every 10 meters, 10 meters pressure increased by 1 atmosphere. So for at 500 meters or 5,000 meters, that's 5 kilometers below a sea surface, and the pressure would be 500 atmosphere. So you must be wondering how these fungi might be living under such a kind of extreme environment. Not only fungi, even bacteria live there, and some other eukaryotes live there. So from there also, NIO has been collecting samples and studying fungi, and there have been many reports, some of them very novel, and which have been cited by, reported by BBC also. This is British Broadcast Corporation. What we have done, we studied fungi associated with tar balls. I will tell what are tar balls. I think I have some 10 minutes. Let me finish in 10 minutes. These are the black things. Here you see these are the things you find on beaches. It's not in Visakhapatnam, not in probably Godavari, Kakinada or somewhere there. You could find this kind of ball-like structure, black ball-like structures, beaches of Chennai, and Kerala beaches, Karnataka beaches, Goan beaches, Maharashtra beaches, and then maybe Gujarat beaches, Mumbai beaches, of course. So these are tar balls. So the common questions we ask, what are those black things? So of course, these are tar balls. Who put them there? Nobody put them there physically, in the sense it is a natural phenomenon, not really a natural phenomenon. When there is an oil spill incident in an open ocean, and especially when the oil tanker passes through and deliberately or unintentionally releases crude oil to the seawater in open environment, sea environment, that's the oil and water becomes oil slick. The oil slick, because of wave action, becomes a ball-like structure because of sunlight and other chemical and physical harsh condition. This slick becomes ball-like structures. So these ball-like structures are tar balls. They are formed in open ocean, and some of them keep on floating, and during suitable time they come to the beaches, nearby beaches. Especially we studied the tar balls, from Goan beaches, because while I was working at NIO Goa, we started this work. So some of them float and come to the beaches. Some of them actually sink and go to the sea bottom. When they come to the beaches, they spoil the beauty of the beauty of the beaches. So what the tourism department do? They actually manually remove it. Okay, they manually remove it and put it somewhere else. Still, the stresses of tarball would be there on the sand. Of course, the tarballs would be floating in the seawater. Nobody does anything about the pelagic tarball or the floating tarballs in seawater. They would be floating, harming the environment. And the immediate man being a selfish animal in some sense. So we would be concerned about tarballs. Are they safe to humans? Of course, these would be unsafe to even marine animals, marine organisms. But important questions as a tourist or as a local person, are they safe to humans? So these are the questions we ask. Some more information tarballs. These are like semi-solid blobs of weathered tarball, solid outside, liquid inside, soft and sticky, brittle and hard, black to brown in color, millimeter to centimeter in size. They are seasonal appearance in India. Basically, you find them in pre-monsoon and post-monsoon season, mostly on the west coast of India, because the oil tanker route is through the Arabian Sea, not from Bay of Bengal. When the oil tanker starts from Arabian countries, goes to Singapore, it uses the Arabian Sea actually, not the Bay of Bengal. Therefore, the tarball is mostly west coast of India's problem. But it's not just a west coast of India's problem, it's world's problem. Here you can see the places from where the tarball pollution have been reported. There are many places, many coastal areas of the world which have been facing tarball pollution because of the tarball deposition on their beaches. What we have been studying the tarball associated microbes. These tarballs are harboring different kinds of bacteria and fungi, planktons. There could be viruses on tarballs, but there are no reports yet. So in this presentation, I'll be talking about fungi. Give me just 5 more minutes or 10 more minutes. Okay. So one of my project students or BTech students who came to work my lab, she went to one of the tourist beaches in Goa, Kandulan beach. She collected tarballs. She isolated fungi. This is how these fungi look in the PDA medium. Then we extracted DNA from these fungal cultures, and when we did the phylogenetic analysis, we could unravel the diversity of the tarball associated fungi. So basically, tarball associated fungi from this particular beach, based on a very minimal sampling, they were found to be belonging to 2 classes, the Mycetes and Saccharomycetes. And this was one of the studies which employed molecular data to describe fungi associated with tarballs. So that was a good beginning. And here is a list of tarball associated fungi. You can go through the list if you are interested from this paper, the ITS-based phylogeny of fungi associated tarballs. This was published in Marine Pollution Bulletin, actually. Similarly, my PhD student went on to another beach in South Goa called Betul Beach, and we found more fungal taxa associated with tarball samples, actually. So here is a list of there are 18 fungal species, or not species, 18 fungal taxa that was reported to be associated with tarballs from Betul Beach in Goa. So the idea here is that we can use these fungi associated with tarballs possibly to degrade the tarballs. We don't know how fast would be the degradation. Idea is to probably use this tarball associated fungi to degrade tarball. Not only fungi, we can use bacteria also. Another thing we can do is we can use mushroom mycelium, and we treat the tarball with mushroom mycelium. At the end of some period, we expect mushroom mycelium to be working on these tarball samples and degrading some of the chemicals or toxic chemicals like pH, and make these tarballs less toxic. And expectedly, the mycelium will become, mushrooms after eating these tarballs. This is just a concept. Here is some more questions that you can refer to later when you get the slides from Madam Sarah. We have a website called www.tarballsin, where you can get information about tarballs, tarball associated microbes. So the important question is, if you find fungi to be interesting, and if you want to be a mycologist, what you can do. that's a good beginning that you have decided to become a mycologist, or you could become a mycologist. First of all, you have developed more interest in fungi and mycology. So how do you do it? You seek information from Google, and you seek information about, whenever you have any question, you seek information from Google Scholar, Google Patents. Now ChatGPT can be used to ask questions about fungi and mycology. You can go to Wikipedia, you can go to some specialized websites like Index Fungorum, Mycobank, NCBA, GenBank. So next you can do is network with like-minded people. There are many WhatsApp groups for fungi or mycology. Join them. You go to social media, go to Facebook, or go to LinkedIn, go to Instagram. Network with like-minded people. Learn together. If you learn something new, share with your feel free to share knowledge, because when you share knowledge, it actually becomes a bigger set of knowledge actually. So it is a beautiful movie on on fungi of Northeast India, by Stephen Exford and Catherine Marciniak. So this is freely available on YouTube. You can watch it actually. And if you are considering that you want to be a professional kind of mycologist, after your BSc, MSc, BSc, you can do MSc in botany or microbiology, then specialize in mycology, then continue your studies towards mycology, earn a PhD degree in mycology. Then probably as you grow up the ladder in the academia, you will be known as a mycologist. This is how the path. But the path is not very straight. It can be zigzag, but you have to be patient actually. Where to start? You can go to Fungi of the Planet Facebook group. You can go to Planet Fungi group, where Stephen Expert and Catherine Marciniak, they actually release this kind of beautiful pictures of mushrooms. Fantastic Fungi is a movie. You can watch the movie. It was on Netflix. I don't know, it's still there. And every year there is Fungi Film Festival. You could register online, and these are online. This is an online event actually. I would like to conclude my talk. I think I'm finishing at 15 hours, 11 minutes. Thank you very much. I want to once again thank the organizers, the Government Degree College, Ramachandrapuram, for giving me this wonderful opportunity, and especially Dr. Sarah and your principal, madam, and the other team members, especially people from other departments, English or physics, and maybe other departments, for coming together and making the fungi as the center of attraction today. I'm sure Dr. Rohit will take this discussion to the next level when he is going to talk about the application of fungi in industry. Of course, I would like to acknowledge CSIR NIO, my organization, my students and collaborators. It is not one person's work; it's a collaborative work of many people. It's a teamwork. And today's audience, you have been fantastic. I would like to have many questions from you. You are welcome to contact me. These are my contact details. You can email me. I'm active on social media. Feel free to connect with me. Thank you very much, ladies and gentlemen.
Thank you, sir, for speaking about the topic. Welcome to the fungi world, where you have presented who a mycologist is, speaking on about how fungi are underappreciated, and you have also outlined the various benefits, classification, and all those things very briefly, with lots of pictorial images of different molds, mushrooms. And you have also stated the edibility of mushrooms and focused a lot on the missing number of mushrooms, with the example of mushrooms to delimit tarbals. So listening to this particular topic, I remembered a few famous words of Gandhiji, who said, I quote, "We may utilize the gifts of nature just as we choose, but in her books, the debits are always equal to its credits." Thank you once again for instilling an interest in many of the participants to search, or I can say research, mycology. Thank you, sir. The platform is now open for discussion by all the various participants. The participants are requested to put their queries either by unmuting their microphones, or you can also ask questions using the chat box available on this particular Google Meet platform. I request all the participants to kindly raise their queries and get your doubts clarified, at the same time improve more knowledge about the topic.
Yes, Harita, please. Harita?
May I know your insights about cordyceps cultivation in India?
Oh, I'm not an expert in cultivation of mushrooms. I'm not sure how to answer that, but you should consult the experts. Sorry.
Sure, sir. Thank you.
Sir, one question from my side, actually.
Yes, madam.
Sir, fungal identification is too difficult because, as you said, those are all looks like that itself. Means all are looking same when we see any fungi. Means especially wild mushrooms, as you said. So, is there any special site for identification? That is for angiosperms identification. Herbarium is there. Digital herbarium is there. In all mobiles, we have one app, that is Google Lens app. Like that, is there any identification app for identification of this fungi?
Ma'am, if you want authentic, reliable identification of fungi.
Yes, that I'm asking.
If you have cultures, you could send to these culture collections like NFCC Pune, MTCC Pune, MTCC Chandigarh, MCC Pune. So they will do the proper taxonomic analysis. There are experts who do it. But if you are looking at the internet sources or free apps or that, I wouldn't recommend such things because you might get some feeble ideas. But if you want authentic information about their identity, it is better to consult experts. But you can always interact on the social media platforms, learn about it. But it is better to always consult taxonomic experts about the identity.
Okay. So when we collect that wild mushrooms or anything, when we identify that, how can we preserve it, sir, just for study? Is there any simple method, laboratory method?
You can sun dry it and just keep it.
Okay. Okay, thank you, sir.
Thank you. Akriti Sharma, you would like to ask me something?
Sarah, madam, there are 3 questions in the chat box. Could you view them once? From Akriti Sharma, Rajarshi, and Aditya Bhatia.
Damodar sir, some questions.
Yes, so I'll let me read. Akriti Sharma wants to know that I want to ask, how did you get too motivated to work in such a diverse field as you work both on taxonomic and molecular aspect of it? I think I was in the right time in the right place. Okay, I did my PhD at the University of Hong Kong, where my PhD advisor was flourishing in his career. He had a big team of students and other postdocs. I'm talking about 2003 to 2007. That was when the fungal taxonomy was embracing molecular data. It was not doing of taxonomy using molecular data became very trendy, very fashionable. Sometimes led to paradigm shift in some groups of fungi. So that way I got into it, and that lab during that in the right time and probably in the right place. At the end of the PhD, 4 years of training, I could understand fungal taxonomy in a better manner, also understand how to use molecular data in fungal taxonomy. Later on in my career, of course, there were opportunities. When I was in IMTech Chandigarh, I worked on terrestrial fungi. I worked on plant pathogens. Then when I plant pathogens, insect pathogens. When I moved to National Institute of Oceanography, I started working on marine fungi. It was not just the diversity, we had to work on some application part. Therefore, we started studying the degradation of the star balls or some other pollutants using. So in the last 15 years of my, not 15 years, 20, 18, 19, 20 years of my experience or working time, I think I got good opportunities. Therefore, when you get a good opportunity, when you get good bosses, and when you get good working environment, motivation is just an addition, actually, just an just a bonus.
Thank you. Thank you so much, sir.
Thank you.
Sir, do you think it is easy to work on taxonomic aspects than molecular, or molecular work is a bit easier than the classical taxonomy?
Oh, classical taxonomy is very difficult. If you want to master one group of fungi, for example, Aspergillus, that will take your 30 years of career.
Yes, sir.
Yeah, but comparatively, you have basic understanding one group of fungi and start applying molecular data. Things looks less difficult, less daunting, and then you get interested and you can do more work. Classical taxonomy.
But sir, you can't actually rely on. Sorry, sir. Sorry to interrupt you in between.
Morphology is very difficult to master. That would take dedicated work of some 15-20 years, provided you get a good job.
Yeah. Yes, sir. So, but we personally, when we get on to the molecular aspects, we get into identification problems, because sometimes we get some good results, and the other time the same results gets means they are something else when we identify it molecularly second time. So, sir, how to rely on molecular aspects rather than leaving the classification taxonomy?
See, basically, taxonomy is for the end users, right?
Yes, sir.
But the end users are in confusion because of this transition period. I mean, fungal taxonomy is still in transition. Okay, it started in 1990s because of incorporation. I mean, started from incorporation of molecular data in fungal taxonomy, ITS and other 18 years and all. Now we are moving towards multi-gene, whole genome, and
Yes, sir.
machine learning and ecological, integrated taxonomy.
Omics tools as well, sir.
Omics, yeah. So basically, you want to test the hypothesis. I mean, for example, test the species as a hypothesis. If you treat that way, if that a species is a hypothesis, there was one species named XYZ, and there was the hypothesis based on morphology. So now you want to test that hypothesis with the new fresh evidence, a new set of data. If that particular hypothesis is still valid, that's fine. Otherwise, you come up with a new hypothesis or new names or a broader definition or a very restricted definition of that. This is how you could treat the advancements in fungal taxonomy. That any taxonomic or any fungal taxon as a hypothesis. Other than species, everything is an artificial group. Right? Okay. This is the only thing which is natural group. Okay. But how close are we with respect to our hypothesis of those groups? Yep.
Okay. Thank you. Thank you so much, sir.
Thank you.
It was a nice presentation, sir. Thank you.
Thank you so much for your participation. So you want me to answer more questions? Maybe we can move on to the next presentation.
Shall I ask you a question?
Yes, madam.
Sir, namaste, sir. Thank you for enlightening us. I'm Dr. Swarupa from GDC, Government Degree College, Doon. And thank you very much for the college people who have enlightened us and make us to know the World Fungus Day. And your elaborative lecture regarding the fungus and many things we came to know the Penicillium notatum we thought actually, and as you said, Penicillium rubens. is used in this antibiotics for the extraction of the antibiotics. And my question is, sir, in this latest research or in your research, generally the fungus association is with lichen, with algae, lichens. Is there any new such associations with any other type of species? And we know that there are some ectotrophic mycorrhiza, endotrophic mycorrhiza, who are in association with the internally or external bodies of the other species. And in new or the latest research, any such associations have you found?
I think recently I came across one report. I didn't read the paper, full paper, like fungi living inside of fungi, something like that. A fungus living inside a fungus. Okay, so that's probably an unusual thing to happen, but we need to dig further. But whatever the association, classical association of lichens,
Yes.
and mycorrhizae. So we are getting better. We are getting better in terms of the understanding. Yesterday there was a talk about bacteria associated with AM fungi. How the diversity and all this matter for the plant growth. So we are getting better with respect to the about the existing associations, symbiotic. Of course, talking about the plant pathology, that has gone to a different level. Now we use whole genome sequence data, competitive analysis and other stuff to understand these associations better actually. But I don't think any newer organisms are getting associated. Understanding is towards like new groups of organisms are having symbiotic relationship fungi. Maybe Rohit could put some light on it when he talks about it.
Yes, sir. So what about this lichens research? What is the status and position of the lichen research?
I think, if you are very keen on lichen, you please talk to Dr. Sanjeev Naika from NBRA Lucknow. He is their expert actually. Madam, I will be happy to connect you with Sanjeev Naika actually. And there are some people in Agarkar Research Institute, Pune. They are also working on lichens, madam. Because I have not done any research on lichens. I wouldn't be having so much knowledge about it actually.
So, what are the advantages of this Marinus fungus, sir?
Advantages are deep sea fungi, as they are living in a very harsh condition. So they could be cultured mimicking the conditions in the lab conditions, in the laboratory. So if you can maintain 500 atmosphere pressure and temperature of 4 degrees in our lab condition, we have pressure vessels. So we can grow them, and if we grow them, we would know the kinds of metabolites they are going to produce. See, to live in such condition, you need different set of metabolites. So we can use such metabolites in drug discovery. But growing such fungi in lab condition would be very challenging. Very few people in India can do it. That's one aspect of marine fungi actually. Secondly, these fungi associated with the marine pollutants, these could be explored further for the bioremediation potential. These are all at the moment lab-based studies. Okay, in the field there are not many products based from fungi for bioremediation, at least in the marine setup actually. Right. Potentials are there, but the realization will take a different level actually.
Okay. Thank you very much, sir.
Thank you, ma'am.
Thank you.
Sir, one question raised by participants, that is Lokesh. Is there any marine fungus which degrades plastics?
We don't know it. We have to do some work and find out. Because there was a report from Japan, which one fungus, Stolopships, is degrading plastic. That probably is a terrestrial one actually. I need to look into that actually. Yeah, but we have a group working on microplastics. We have isolated fungi from microplastics in another study. And there is a confusion. What we are isolating is the things which are outside the microplastic palette. So that could be dust, that could be sand particle. How do you really make sure these are microplastic associated? So when you say associated, it could be very loose interaction. So we should be very careful about such. And after that only we can go for degradation studies. I think we are at very beginning stage, very primitive stage, with respect to degradation of plastics using fungi. I assume that fungi are quite capable of doing such things. Unless and until we produce evidence, it is very difficult to believe actually. Thank you.
Sir, for taking so much of pain but answering all the queries with such a calm composure. We are really happy to have you, sir. We are thankful for all the useful insights you have provided to all our participants. Sir. Thank you, Damodar Shanaivar.
Thank you
sir.
Thank you once again to the GDC Ramachandrapuram and Dr. Sarah Madam. Thank you so much. Yeah.
Now I would like to request Srimati G. Sushila Garu, lecturer in chemistry, who is a learned academician, having a humane spirit, a very hardworking individual, to brief out the profile of Dr. Rohit Sharma ji, our second resource person. Over to you, Srimati Sushila Garu.
Good afternoon, one and all. This is Shushila, lecturer in chemistry. It is my pleasure to introduce our guest speaker today. He is Dr. Rohit Sharma. He is eminent mycologist in India. He completed Bachelor of Science to Master of Science in different institutions present in Jabalpur. He completed Doctor of Philosophy from Rani Durgavati University in Jabalpur. He got so many awards, especially he received University Gold Medal from obtaining highest marks in MSc Botany from Rani Durgavati University, Madhya Pradesh. Also received first prize at Junior Scientist Award from Society for Basic and Applied Mycology, Department of Biological Sciences, Rani Durgavati University, Jabalpur. He had so much of research experience in exploration and collection of fungi from natural habitats and endophyte, identification of microfungi using polyphasic method, and isolation of fungi using enrichment techniques. At present, he is working in Professor of School Sciences and R&D Coordinator, SAGE University, Bhopal. His work mainly focuses on taxonomy and systematic of fungi. He is also working on biodiversity of fungi from different niches across the country, from Sikkim forests to the Biosphere Reserve of Chhattisgarh. At present, his research interests are in role of fungi in hardening of tissue cultured plantlets and conservation of endangered plants of Panchmarhi forests. Thank you, sir. Thank you, one and all. Sir, sincerely, we are waiting for your session, sir. Thank you.
Thank you so much, Srimati Sushila Garu, for nicely briefing out our resource person. I would now like to warmly welcome resource person and facilitator, Professor Dr. Rohit Sharma ji, a prominent researcher and active member of various scientific societies, a well-known reviewer, a splendid academician from ISC University, Bhopal, Madhya Pradesh, who has accepted our invitation to present his topic, Biotechnological Potential of Fungi, in the webinar. We are grateful to have you as a resource person of today's event, sir. आपका हार्दिक स्वागत है, sir. Over to you, sir.
Yeah, thank you, Dr. Sushila, for the nice introduction. Before I start, I'll reciprocate Dr. Shnoy's sentiments for congratulating the team of the Government Degree College for the organizing this World Fungus Day. It's very heartwarming to see all these large number of participants to participating in the webinar. Before I start my actual talk, I'll just want to just comment on what on the Dr. Shnoy's presentation. That even I observed the tar balls in the beaches of Alibag. I had been to several of the beaches of when I was in Pune. I visited several of the beaches in Western Ghats and of the coastal region. But when I visited the Alibag, and I tell you what Dr. Shnoy was telling is correct, that it's very irritating. It is very irritating when the tar balls are there in the beaches and you are not able to. It's very sticky and it's irritating kind of thing. Yeah. So I'll start to share my screen. I think means do I have the permission to share my window? Yeah. Is my presentation visible?
Yes, sir.
Mhm. Yeah. So is my presentation visible in full screen?
Yes, sir.
Yeah. So, although the 2nd of October is known for Gandhi Jayanti as well as World Fungus Day, but the day is also the beginning of the announcement of special things that will be coming in a few days from now. And that is for this thing, many of the scientists they have to get up very early. So, as it Professor Harvey has already told in earlier that. He was very first angry at being disturbed at a call at late night on 5th of October 2020 that quickly changed when he learned that he had been awarded as the Nobel Prize. So it's the best alarm clock that he had heard, and you get me right that it's the Nobel Prize announcement that will be beginning from today to up to 9th. So just to brief you all, the Nobel Prize.
Sorry, Rohit, you are not on full mode, screen mode. अभी तक नहीं.
Oh, okay.
Our present from PDF, please.
Sure, I'll do that. Is it now full screen?
No. Rohit. Just start using. Can you use your PDF file if you have?
Yeah, I can use my PDF file. Or is it okay if I share it to you and then it can be shared, because there will be some slides that will go back.
Give it to me, yes.
Yeah.
You can WhatsApp me.
Sure.
Once again, somebody says F5.
F5, okay, I'll just check.
It's working now. Just hide that notification. It's working now. Just hide that notification. Thank you. Great. Great. Thank you, Lokesh.
Yeah. So the Nobel Prize, just for the information of the students, the Nobel Prize was set up when a businessman and entrepreneur Alfred Nobel died and left the majority of his fortune to the establishment of prizes in the physics, chemistry, physiology, and for medicine and literature and peace. So he has stated that the prizes should be awarded to those who during the preceding year shall have conferred the greatest benefit to the humankind. And most of us must be knowing these two persons, Dr. Venki and Abhijit, who has done extremely good work and were awarded the Nobel Prize, although they are the Indian origin citizens. Many of the talks that I've been taking up with the students in small schools, orphanages, and kids with special needs. The first point that I have mentioned in the quorum sensing, one of the students has asked at one of the center was how microbes talk between themselves. He asked when we people can communicate between us, then how these microbes talk. And that is one of the concept that we know is quorum sensing. And another student once had asked me that how do the microbes breathe if they are present inside our gut or inside our body. So, and there was a Nobel Prize in 2019 for the discovery of how cells sense and adapt to the oxygen adaptability. So you see, the point I want to make is the ideas comes at even anybody can get an idea. Only thing is the person who is getting the facility may be working nicely and they may be taking the science ahead. But ideas are not bounded to any person, but anybody can get an idea. Even students of 6th or 7th class even can get it. And that's what Professor Serge has said, that the wealth of a country comes from the minds of its people. Coming to my talk, that is the bioperspective of fungi or the biotechnological aspects of the fungi, I'll take you to a journey wherein we'll discuss some of the applications that I have been working on, and in general, they are known. And some very interesting things we'll be discussing over it. We need to understand that fungi has a lot of potential. Fungi have a lot of potential. Earlier, I was working in a basic taxonomy in the out of my 14 years of career. Around 10 years, I had been working on the taxonomy of the fungi. But later on, when I realized that Dr. Ranade from Agarkar Research Institute, he has forced me that you should also work on applications of fungi. But I was reluctant to do it. I thought that no, I am only a taxonomist, so I cannot work on practical aspects. But he should just try it. And when I started working on fungi, applications of fungi, I came to understand that they have amazing property and they have no limits. They will surprise you what they can do it at every level. Before I start, I'll thank and receive the funding from the DBT and other agencies, as well as my contributions of collaborators, colleagues, students, technicians, and developers. Some of the applications we are well aware, like the aspergillus is there, plutus and agaricus are there, the edible mushrooms, Saccharomyces is there for the bread making and cake makings, we are well aware. Aspergillus and many penicilliums we know for the cheese making kind of thing. But some of the applications have been little bit people are unaware. One of the things are the ectomycorrhizal mushrooms. One of the madam was making a point. Yes, ectomycorrhizal mushrooms have an important aspect while the plantation of the trees are concerned. Some of the ectomycorrhizal mushrooms are used as an inoculum for the in the nurseries by the forest people for the plantation of the ectomycorrhizal trees kind of thing. And some of the mushrooms are poisonous also, but they are also using some of the important secondary metabolites for manufacturing some of the other some of the things. They are hyperdiverse, as Dr. Sunal was mentioning. They are hyperdiverse. There are a very large number of things present. Many, some of them we have searched, able to describe them, but many of them we have not been able to describe. So it's a huge gap. It's a huge area where we can, the students or the participations is necessary. More and more taxonomists and people are required to work in this area, because until and unless you have the culture you cannot work on the its exploitation or the its working. So we need more and more people working on the diversity as well as working on the taxonomy. Because even if you can obtain a fungal cultures, until unless you know its identity, it's of no use. So we need to understand that also. You know that if I ask you which is the most dirtiest spot in your home, you will definitely give me a point that okay, my toilet or my bathrooms must be the most dirtiest place. But in one of the study, it has been found that the kitchens or the kitchen sink is the most dirtiest. A whopping 70%, 5% of the dish sponges and rags were contaminated with the coliform bacteria, which is an indicator of the possible fecal matter. So these are some of the things which we consider that where the microbial diversity or where the things can be obtained, and that shifts our concepts from where we can look into it. So we need to understand where these things are. Where we can look the means we can say unique habitats, where we can look into unique habitat. So these are some of the unique habitats. Instead of looking into the bathrooms, we should look into the kitchens. Instead of looking into normal habitats, we are looking into the unusual habitats like mobile phone surfaces. I do not mean that they live on the mobile surface. It's not their substrate, but it happens to be on the living on the surface, adhering to it. Okay, they are not feeding or living on these kinds of microchips kind of things. So unusual habitats include the meteor impact lunar sites also, which is hypersaline. More than 9 is the pH of the lake, which is formed by the impact of a meteorite thing more than 40 or 50,000 ago. So these are some of the unusual habitats where one can look into the organisms, and since they are living there, so they will have this special ability. When they will have this special ability, so that will be of use to you. It's a 5-kilometer diameter kind of thing, circumference, and around 2.5 kilometers of diameter is the complete diameter of that particular thing. Working on fungi is not very easy. So the day when you are going for a sampling, the day may start with a smile, and as the day passes, you may have to have a lot of tough time also. And when the day is coming to an end, your faces will be like this. So it's not very easy to work on fungi. It may take a whole day to find some kind of good samples, good isolates samples which can give you good isolates. Mucormycosis, Dr. Shnoy was talking of that. I agree, mucormycosis has been an important thing during the corona, and people, it gave, I should not say recognition is the right word, but I say that it gave popularity to the fungal taxonomies, because when I was in Pune, so one of the NABL laboratory has given the responsibility to identify the fungi of mucormycosis infections, but they did not have the expertise. So they were coming to me at the at that time I was at NCMR Pune. So and immediately we can say DNA isolation, PCR sequencing take a long time, but the morphological identification was serving the purpose at that time, because the doctor only wanted to know which group of fungi it is, whether it is an mucor or the rhizopus or the aspergillus or what kind of, so that they can decide on the antifungal treatments at the earliest. And I was surprised I could isolate even one of the neurospora also from one of the samples. And because neurospora in general is not known for a pathogen, but I could isolate that also. And I was surprised, and it was quite evident that because the patient was a fruit seller, and we know that the neurospora normally occurs in the kind of fruits kind of thing and those kinds of things. So these are things. So people are also now, we shifted some focus, although I could not continue on this aspect, but there was a necessity for, just like COVID shell, there is a necessity for a rapid identification of fungal kit. Is a necessity that we are in discussion with some of my collaborators to work on it. Antibiotic resistance, or in our sense, antifungal resistance is developing it's considered to be a hidden kind of pandemic. So in case of our fungus, Candida auris has been considered to be multi-drug resistance, and people are working over it to and this kind of thing. So these are a lot of aspects that need to be understood for the kind of thing. And as Dr. Shnoy was mentioning, that first antibiotic was discovered from fungi. So these new fungi are also important so that we can have some of the new antibiotics also, because antibiotic resistance are developing. So we need some isolation of some fungi from unique habitats so that they can be used for that thing. At present, most of the people are working on endophytes. So most of the fungal endophytes that has been isolated from plants leaves are showing some potential for the antibiotics potentials. So we have isolated some of the fungi from the Sikkim forest from the plant endophytes, and they have been showing some possibilities of good responses when we are testing them at the standard cultures. We have procured some 7-8 standard cultures which are being used by the pharma companies as well as pharmacopoeia. These cultures, standard cultures, we are testing, and we are finding some of the good results into it for that thing. So human microbiome, including fungi, has been an important aspect for most of the microbiologists and microbiologists nowadays. And these are being also studied, including like in Candida, most of the our body flora of Candida are later on when the immune reduces, the same Candida is becomes pathogenic. So people are working on that thing also. Fecal microbiome transplant is the near future, that is some of the hospitals in Delhi as well as in Beach Candy Hospital in Mumbai, and these people are also working into the fecal microbiome transplant. Like an obese person, instead of going for a surgery or kind of thing, they are going for fecal microbiome transplant for this kind of thing. And it includes, I'll tell you, it includes fungi also. So related to it, we have been working on isolation of some of the important fungi which can degrade cellulose quite fast from the gut of termites. So we have been isolating some of the fungi which are living in the gut of termites, which, as you know, the termites are very fast degraders of the cellulose. So we are isolating some of the fungi, taking clue from this kind of aspects. So nowadays, these fecal microbiome transplant capsules are where they have ultra filtered the remnants, and only the microbiome is there for this kind of thing. The idea is that they transplant it into the gut of that thing, and slowly, it's a 3 months process wherein the fecal microbiome or the gut microbiome gets replaced by other microbiome, which reduces the obesity. Yeah, NASA has been also keen on microbes. They have been working on fungi and bacteria both at their space stations. Also, they are exposing these microorganisms for the UV radiations and other things. And because there the radiation is quite high, 15 or more than, I think, many times high is there. So they have been working. They have even isolated a novel fungi from their facility. You need to understand that all these satellites, they are what are sent to the space, they are completely sterilized. You will ask that what are microbiologists doing in the NASA thing, because they have to completely sterilize the thing, because no microbes should, or no living organism should live into the Earth surface or the Earth atmosphere. So they have been studying. So they because their facilities very stringent sterility is there, their thing is very clean. So even there they have found some of the microbes, and they have isolated some of the fungi there, and they have described they have come to be novel, and they are very dangerous because when they are living are found to be living inside the NASA high safety and high very means clean environment. So they are very dangerous because they are able to sustain their procedures kind of thing. So two of the Indian scientists like Kasturi Venkateswaran and Parag Pran are both working out there. At present, probably they are working on the Mars Center, also Mars mission, Jet Propulsion Laboratory in California. So coming to the second portion of my talk, I will be discussing on some of the practical applications that we have been working on as the waste treatment, composting, micropackaging, and other research and innovations. To begin with distillery waste, So distillery waste, you should understand that it's distilleries are a subsidy or this kind of an industry which is parallel with the sugar industries, wherein they produce lot of molasses. Sugar industry and these molasses are treated into the distilleries to produce alcohols of the industrial as well as the drinking standards. And even in Maharashtra, there are 200 industries are there, and in all over India, around 400 industries are there. And they produce for production of 1 liter of alcohol, it needs 4 liters of fresh water. So you can imagine how much of fresh water are needed. But when the waste comes out, it's a very dark brown in color, which is rich in melanoidin kind of thing. So here you can see that this is the dark brown color kind of distillery waste that is coming out of the thing. Here you can see that the distillery waste. So when it comes, it's two kinds of things are there. One is pre-methylated spent wash, and one is post-methylated. So in the pre-methylated means it's directly coming out from the distilleries, and in post-methylated it is has to undergo the anaerobic digestion by the microbes, and then it comes out. But both are brown in color. All the COD in body is quite reduced in the post-methylated thing, but the color is quite dark brown in color. So it is very bad, and at present there is no technology available to treat this kind of thing at this scale. So what they are doing it, they are storing in large lagoon kinds of thing, and then they are using it for composting also, and then they are discharging into the rivers in the rainy seasons and just without treatment. So it contains whole lot of compounds, including phenolics, melanoidins, and other things. But the major one is the melanoidin, which consists of the around 80 to 85% dark brown color presence is because of the melanoidin. So there have been several technologies. People are using it. Some are using sedimentation. Some are using UV light also. Many technologies have been used. Anaerobic is the majorly been used, but in anaerobic, what is happening is they're only reducing the COD, BOD, but not reducing the brown color. So that is why we took this project at the previous organization, wherein we screened several fungi for that thing. And for this, we took some of the important fungi, that is the wood degrading fungi, because many of the wood degrading fungi, they have the capacity to produce lacase enzymes, and those it is known in literature that those fungi, they produce lacase enzymes, they have the capacity to degrade this kind of thing. So you can imagine here that the COD and BOD is around 6,800, and the pre-methylated is 8,400, 84,000, and the biomethylated, that is the post-methylated, it reduces to 34,000 or 11,000, that's COD, BOD. And the color, it is, if you can see, its color is brown, brown, brown. So there is no change in color kind of thing. And it's quite high COD and BOD in the waste that is coming out. So we screened some of the fungi, and you can see on the rightmost kind of thing, the control, you can see it's dark brown color, and where the fungi was used for the treatment, it converted into quite yellowish or the white portion media. This was the preliminary screening done. Then later on, we screened more than 10,000 fungi into it, and after getting a primary screening, then we did the secondary screening. And in the secondary screening, we identified two strains. That is, the PF9 was identified, one of the important strain, which was showing around 89 to 90% decolorization capacity of this thing. So it is known as spent wash. Many places you must be seeing the word that spent wash. So spent wash is the waste coming out from the distilleries. You can see here the right hand side, it makes it quite yellowish and quite light in color as compared to the dark brown color, which is the original thing, in just 15 days. And when we repeated it in the second time, the number of days, since the inoculum was already generated, the number of days reduced to about 7 days. Okay, so it's very significant declaration taking place. We also tested them for the lacase enzyme, and all the strains, the two strains were ABTS. Through the ABTS substrate, we tested that they were lacase positive. We also did the genome analysis for both the strains, and it was found that the oxidoreductase enzyme scaffold was quite There were several dominant in there, and so we can find that because these oxidoradases are the enzymes which are responsible for the decolorization or the biodegradation of the pollutants like many of the alkaloids and kind of thing or xenobiotic compounds. So that's why we are interpreting this kind of thing also. And once we treat it, you can see on the right kind of thing the pre, post, and post. You can see PF9 when it is treated, the BOD was came to 1800. Here the BOD came to 1600. Here the COD reduced to 1100. It reduced to 10,000. So significant amount of reduction in the BOD and COD by the both the strains, and as well as the color came to about one kind of unit which was tested, and even the total solid dissolved solids were also reduced a lot, kind of thing. So there was significant reduction into it. Thereafter, we also tested the degradation of melanoidin, and we purified the melanoidin and tested in the HPLC, and it was degraded into two compounds into it. And these two compounds we need to identify. Although we are working on it because we need to also identify whether these two compounds are not more toxic than the melanoidin. They should not be toxic to it. So we are on at this stage of working at it, but we are surely clear that the melanoidin has been broken down into the other components, kind of thing. So we have also combined this biological method or the microbial method with the UV light, and then we could achieve up to 90 to 95 kind of % of decoloration was achieved using it. So these kinds of things we had tried to design. So we had not reached up to this level, but this was the thing where we can design this kind of microbial growth can be done, fungal growth can be done, and the water can be put into it and can be there for 5 or 7 days, because you understand here the number of days. I'll go back. I'm sorry. I'll go back to my previous slide. Here the 15 days. It is not significant. It's too big. It's too big because I am talking about 40,000 to 50,000 liters of the waste that is being generated, and we cannot hold this for 15 days. So we needed to reduce it. We needed to reduce the amount. That is why the reduction in time is more important. So we were able to reduce up to 7 days, kind of thing, because that is retention time is very important in this. The challenges we are facing is that we need to extrapolate up to up to 40 or 15,000 liters, and we have to manage the other environmental factors into it. And definitely, it's part of the scale-up activity. We need some kind of testing 1 or 2 or 3 years for replicating this thing at the industrial scale. Poultry waste is another thing. We have tried to develop a poultry waste. One of the industry wanted to convert their poultry waste into the compost. So you know that the poultry waste is very high in the nitrogen, ammonia, and other things. It cannot be directly used into the field. Otherwise, the crop will burn. So they wanted to use us to use it. And as per the literature, you can also see that many of the fungal mycelia, when mixed with the poultry waste, they convert them into a usable composting kind of thing. For this, one of the client has approached us for this thing. So we tried to mix. We generated the mycelium at a large scale. Here we use the mushroom mycelia of Floritus in a large scale, and we also compared it with the bacteria and with other fungi also. 3 replicates we have taken, and and one was controlled without any inoculation. And we could find that there was significant difference between the fungal mycelia doing the job quite good. So first we inoculated the thing, mushroom mycelia, and then you can see the thing over there. And slowly, the temperature start increasing and at about 50 or 60 degrees, so that you can feel that the microbial activity is taking place, kind of thing. But a regular monitoring, you can see the right hand side, the time is there. So every day 2 p.m. So we maintain the every regular timing at kind of thing, and then we could achieve the compost composition of a CN ratio of at least approximately 12 to 12.2% it was reached, kind of thing, and we could achieve the objectives of the client. And now he's ready for his because nowadays fungal or the microbes are playing a very important role in the circular economy. This is part of a circular economy. So the poultry waste should not go as a waste, but it should be used as a composting, and a secondary industry is established with the waste. So that is playing a lot of role. Another is the micro packaging that is very close to my heart. I am working very keen on it to work on it. You see, the fungi, stubbles has been creating a lot of problem, especially in Delhi or nearby area. You must have heard that every winter they face a lot of problem, and there are a lot of industries that can be established to help that. Apart from the composting that is can be done, or the mushroom cultivation, or the animal feeds that people are using, micro packaging is an important field that I think it can give a solution to the stubble burning or these kinds of things. Only thing we need to have a kind of focused orientation of the government, and it can solve. It can act as a replacement to the thermocols and the styrofoams. So for micro packaging, what are doing is we are trying to use the wheat straw or the paddy straw and inoculating the mushroom mycelia, preferably lotus, and then molding it into different shapes to achieve these shapes. So one can achieve the shape of a pot or a packaging for the bottles and other things one can achieve. So it's very interesting. It's very hard, very tough. It's very hard. It can be used. So we have been trying to make bricks. We are making bricks also. We are making pots also using it in the initial. These are all preliminary experiments. Now we have improving on it, and we have improvised over it with the help of some of the standardized conditions for heating and incubation. And we are also working on the plates, generating of plates also. So there are some of the things that micro packaging can bring into it. One of the important aspects that many people know is the drugs from the endophytes, and this is the Taxol that is most popular thing that was it was a plant endophyte fungus which produces this thing which was isolated from the bark of the plant, that is Taxus. So there have been some of the very good examples. They have now been kind of unique in their thing, because there is no replacement to this kind of drugs that has been developed from fungi. Bioethanol is again a very important aspect. That is, we had worked on the lactase enzyme screening by one of the industry of in Pune. They are the largest bioethanol producers, that is the Praj. So we had screened fungi for them, and I'll tell you, fungi are good lactase producer. So they are being used for degradation or degrading the sugarcane bagasse and wheat straw and other things, and then they are producing bioethanol from it. Yeah, mushroom cultivation is important. Everybody knows, but it has been given a new kind of life by some of the organizations like Nuedo or the organic octrines. They have been working towards new packaging kind of thing by the help of mushroom kits, or by the help of kind of Ganoderma or Cordyceps. So people are doing a lot of cultivation and good. Even some of my senior citizen family members, they are also taking once in a week Ganoderma for immune, because after Corona you need some kind of immune boosting, and they are helping very nicely, at least readily available. Because although some of the like your moringa and other things are also antiviral properties, so they are also similar to that is available in powder form. So now these things are also available in powder forms, even in tea forms also. So this mushroom kits are now a new concept that is very good. You can take your home, just spray water for some time, and then you'll get fresh mushroom at your home. Other important aspect for this thing is the perfumes. Yes, some of the people are isolating important perfumes from the mushrooms because they have the fragrance into it. So a specific compound they can isolate from there. So they are also important aspect. Apart from that, they are also important for the colors, dyes. Many of the mushrooms are being used for extraction or the production of the dyes, as well as things, because they are very producing. Eco-friendly dye is available, and these, if it is the mushroom is edible, it can be used for your ice creams and all those things, and any edible things kind of thing. So even we have tried to make some papers out of mushroom by some of the graduate and under PG and UG students in their laboratory experiments or the project-based learning. They tried to make paper out of some mushrooms. So it was good. It was fun making it, and we were able to kind of thing. But we are in a process of testing its tensile strength, blotting thing, every other parameters we are in the process of making. testing it. So I can talk, and we all, means everybody knows some of the applications like biofertilizer, biopesticide, tofu is made of soya sauce, composting, cheese making. So it's unending. So application of fungi is unending. They can go on and on and on. But I'll just like to show you some of the unique application of fungi that people are testing for the road making, and you'll be surprised to know that some of the places where there are landslides are happening and where the roads get in the hilly region, people are trying to inoculate the roadside with the help of straw with the mushroom mycelia, and see, because mushroom mycelia is known to bind it, kind of thing. And they are then, when they grow nicely, the mushroom mycelia goes inside the soils and they bind the soil. Once they bind the soil, then there the soil erosion is very less. So people are even testing into it as a micromatting to the edge of the roads, creates a filtration interface, preventing the soil erosion. So they are even trying to microrestoration using the abundant roads for that. So fungi play an important role in biodegradation and bioremediation also, and including fungal enzymes. And fungi and other microbes can provide solutions to many man-made problems, especially present environmental problems also, and sustainable development. Just to add to my talk some extra points to convince you all youngsters what need to be done. So without the love of research, mere knowledge and intelligence cannot make a scientist. That was the thought of Madame Curie. And you know that we are facing a climate change at present, and that's the most important threat that we are facing all over the world. Even in recent times, Nagpur has faced a lot of cloud bursting and were affected with it. And many of the cities of our country are affecting. So forest fires become quite common in Australia and even in US forests. So it is being increasing year by year, and now the climate change has reached up to the doorstep of each and every person of the world. You can know that which is better. This is better or this is better. Definitely, I am not saying 99. I am saying 100% of the participants will agree that this is better. Okay, this is better or this is better. We do not want our elephants to be a part of our garbage, but they belong to this natural place. And why us? I am talking that why us, or why us will be responsible. Because even though our DNA is similar to 98% to gorillas and 99% to chimpanzees, humans will be held responsible for the degradation of our planet and destruction of the planet, not the chimpanzees or the gorillas, because we are more intelligent and we are in the control of the planet, not them. So we need to shout out loud and raise our voice towards the conservation of biodiversity, because there is no other planet to touch down. And you know, the planet is very beautiful out there. It's called as the blue planet, and a single one there. Thank you.
Thank you so much, sir, for such a vast, detailed, enlightening lecture. Your topic, bioprospecting of fungi, was so informative. You have surprised us all by pointing us with unusual habitats both at home and outside, which teem with germs, even like a simple device like a mobile or even the kitchen. You have also dealt with in detail about various aspects of physiological diseases and how the drug resistance has developed during the COVID period due to excessive use of antibiotics. You have also showcased your work and also explained the importance and association of microbiologists with NASA, even space research for identification of microbes. You have also highlighted established standard protocols and grading systems, treatment strategies used in treating spent wash, primary and secondary screening for decolorization. And lastly, you have also pointed out to the usage of fungi in decomposting. You have brought out mushroom cultivation, their applications, and brought out various images of work experiments which you might have conducted in labs, and also spread the micro message of how fungi can be used for restoration of even abandoned roads to bring about a difference in bringing forward environmental sustainability, and also stating the unending uses of fungi. We are grateful to have a wonderful resource person today like you, sir.
Thank you.
Thank you, sir. Now, the platform is open for discussion. It invites queries from the participants for discussion. May I have any participants who have queries? You can use the chat box, or you can also unmute your microphones and put in questions. I hope the participants are ready.
Dear participants, you please unmute yourself and please ask questions.
Hello. May I ask?
Yes, Rajeshri, go ahead.
Thank you, sir, for the wonderful presentation. And I want to know about the available resources to learn the in-depth of classical taxonomy, such as for certain group of fungus, how to identify them by seeing their morphology and microscopic features. Is there any available online resources or some in-depth resources to learn them, not just identify, but their principles of classification and learning them?
Yeah, Rajeshi, normally what happens that as Dr. Shnoy has also pointed out during the earlier questionnaire session, that many culture collections, what is happening that culture collections are organizing several workshops related to taxonomy. So once if you can attend it, so they will be you will be able to learn both the classical, that is the morphological based identification also, as well as the molecular based identification also. Because number one, number two, we should understand that our most of the this fungi are divided into three non-formal groups. That is, the one is the yeast, one is the microfungi, and one is the macrofungi. And all three morphological identification species concept are different. So for the yeast, it will go as kind of, you have to do API test, you have to do the sugar and the enzyme test. For the macrofungi, you have to observe the morphological characters of the stem, everything kind of thing. Then for the microfungi, there are different things. You have to observe the spore structures and other things. So all the three groups follow all the three different kinds of morphological identification features. So I'll suggest that based on it, try to follow some of the workshop that is available for it. As Dr. Shnoy has pointed out, MTCC, NFCCI, NCMR at Pune, all these regularly once in a year or twice in a year conducts it. Or if at some point, if you feel that my help is required, so I can help you out regarding kind of morphological identification for particular kind of genus or kind of thing. Or you can even contact some of the taxonomists, good taxonomists around in our country. They will definitely like to help you. Because you see, the important thing is for morphological identification, why people are not able to do is the lack of resources, as you said, lack of resource material, that is monographs and all those things. So until unless your laboratory or your lab has the morphological manuals for the morphological identification, whether it is for the alternaria, fungi, fusarium and all those things. So their manuals are available, kind of thing. And there is one good publication, but I do not know whether some of the publications are out of thing. But this studies in mycology, they regularly come up with new what is the species concept going on in particular genus. So they're regular, so they can follow that also.
Rohit, I want to add
so much.
Rohit, I want to add a couple of things.
Yes, yes, yes.
Rajeshri, I know you asked the same question during my presentation. Rajeshri, can you hear me?
Yes, sir. The same question.
Yes. Rohit has given sufficient information. Just a couple of things I wanted to add. There is one website called Fifth Kingdom. It's a book written by Bryce Kendrick. Okay. That website has got almost free content. Okay. And if you go to their reference section, there would be many references that you can search for. Number one. Number two, you can go to Mycobank website. Okay. So that Mycobank gets updated almost every day about all kinds of fungi that we are reporting or reported. So Mycobank and Fifth Kingdom, Rajeshri. Okay.
Thank you very much, sir. Thank you both of you for your detailed answers. Thank you very much.
Thanks, Rajeshri.
Hello? Yeah, yes, please go ahead. Please go ahead.
What is the significance of the concept of micro restoration? Micro restoration. What is the significance?
Myco
restoration.
Micro restoration, yeah. Micro restoration, you see, recently there is a grant proposal submitted. You see, the concept, I think the slide that I have shown is very significant, because we should understand in the past couple of years, we have been witnessing a lot of landslides in the Himachal Pradesh region because of the kind of overcrowding of the tourists, pressure to these hilly areas. Number two is construction and those things. But at the same time, we should understand that we are losing a lot of biodiversity. We only focus on the above ground diversity, but we are not focusing on the below ground diversity of, that is, I mean to say, of the microfungi. That are the main binding strength of things. We only focus that, okay, grasses and trees are the binding things. No, fungi are the thing. The above ground diversity is dependent on the below ground diversity. So the more the fungi are there in the thing, the more the above ground diversity will be there. And they are there for kind of things. So for kind of micro restoration thing, definitely they can play an important aspect. There needs to be a pilot studies to be it, and I think recently I had seen some grant proposal in some good university in Europe kind of thing, where people have trying to develop this kind of thing, where they can be inoculated so that the fungi is there, and so the landslides are less, and that holds a great importance, that I feel. Because soil erosion, you see, we also understand the soil erosion in agriculture field. Also, the topsoil is going away. We are working towards soil health. But we need to understand the microbial health of the soil. We are not focusing on that. So we need to understand the fungi. It's not that because I work on fungi, but I think that fungi has immense potential, and they are being neglected many times. Some of the things that fungi can do, not even bacteria can do, which are more practically used or applied kind of thing.
Thank you. Thank you.
Yeah.
Any other questions?
May I ask?
Yes.
Okay. I have read that there are role of microbes such as bacteria and fungus in formation of rain droplets. want to know more about that. And I just not a question, but a piece of information for the audience. I want to add that there was a question about fungi living inside fungi. I have read at a certain place that algae can also read inside fungal cell, and it is seen in Geosiphon pyriformis. Is there? Can you elaborate this kind of phenomena? Thank you, sir.
Rajshri, means for both your questions, I will not be able to add much because I am unknown about the fact of the rain droplets. Number one, if anybody else can answer that question, I will be happy. And as far as second is also concerned, I am not aware about it, but I will definitely add to it that mycovirus. There are many viruses that live inside the virus, so that is a complete separate field. So there are many viruses they live specifically inside the fungi. And still, like when I was studying on, I worked a little bit on endophytes. So when I work on endophytes, so we have found that the fungi are living inside the plant leaf, but there are many bacteria also that is living inside the fungi. That is also some of the reviews I have listened. But mycovirus, I know that's a complete separate field, and people have studied extensively over the viruses living inside the fungi. But as far as the both the concerns that you have mentioned, I am sorry, I am unable to answer those.
It's okay. It's okay. I want to know one more thing, that it seems that some fungi, especially microfungi, grow in nitrogen deficient culture medium, such as Ashby manito agar, or sometimes they survive on such kind of culture media. Could it be possible that some kind of bacteria, nitrogen fixing bacteria associated with this fungus is making them able to do so?
Yeah, in this specific case, I cannot say that, okay, this bacteria will be doing it, but what is happening is two things we should understand, that when an fungi is living in a low nutrition thing, there are many oligotrophic. I am now working on some kind of samples which are oligotrophic. So they only grow when our media is 1/10 or 1/15 kind of thing. When the nutrition is rich, they won't grow. So there are some of the fungi, or even bacteria is quite common, but in fungi also there are places where oligotrophic is nature is there, so they need very less nutrition to grow, kind of thing. But as far as you pointed to, I'll just add to it. I do not know whether nitrogen fixing bacteria is helping, but in another case that I have studied is in Canthurellus cyberis. It is an ectomycorrhizal mushrooms with pinus. It is found in European countries, in UK and other places, and Sweden also. Uppsala University in Sweden, Dr. Eric Daniel, he has worked extensively on it, and he had showed that when the basidospore of Canthurellus cyberis is inoculated into the PD or MEA, it won't germinate. Basidospores, when inoculated into or spread over the PD or MEA, it won't germinate. But the same spores, when inoculated into media wherein Pseudomonas aeruginosa is growing on, efflorescens or aeruginosa species I am forgetting, but Pseudomonas species growing on, it will germinate. And the same bacteria was isolated as an from the Canthurellus cyberis species also. So then they concluded that this bacteria is releasing, triggering something to break the dormancy of the spores of the Canthurellus cyberis mushrooms. So,
thank you, sir.
You are, if you have to test thing, I do not know, but you can see the literature, or you should yourself go ahead and just try for it, whether it is happening, kind of thing.
Thank you very much, sir.
Yes.
Thank you so much, Rohit Sharma ji, for giving an immediate response to the queries asked by participants. Yes, sir.
Ma'am, if I have a permission, if I can just answer to what Dr. Sara has asked in the previous talk. So, am I allowed to do that?
Yes, sir. Yes, sir. Sure, sir. You can go along.
Yes, sir.
Yeah. So, is my screen visible? Is my screen visible?
Yes, sir. Yes, sir.
Yeah.
It's
So,
visible.
Dr. Sarah was asking how to dry the mushrooms. So when we used to collect the mushrooms, we used to collect it in the newspaper package because it is having aeration and it can be dried easily, and also the bamboo baskets kind of thing. So it is very good to collect it. As far as drying is concerned, so you can dry the mushroom using this kind of dryers are available. So you can use heat convectors also for drying the mushrooms. And for preservation, as Dr. Shnoy said, you can dry the mushroom, pack into the sealed polythene, and just add 1 or 2 naphthalene balls to avoid mites. You are using naphthalene balls, so mites will not eat the spores also, as well as the mushroom fruiting bodies can be, instead of bottles, you can use sealed polythene also. And instead of this kind of dryer, you can use the heat convector for drying the mushrooms.
Okay, thank you, sir.
Thank you very much, sir. It was a really wonderful and enlightening session with a detailed explanation. Now, I would like to call upon the Vice Principal of our college and the in charge of the Department of Mathematics, Ms. Y. Padmaja ma'am, an academician for an entire decade. Her fervor for teaching, her service-oriented nature, and the charisma she reflects with her spirituality are all amazing. I request you, ma'am, to introduce the resource person, Dr. A. Srinivas Rao Garu, by presenting his profile to all the participants. Over to you, ma'am.
Thank you very much, ma'am. Namaste to one and all. It's a great pleasure to associate myself in this great day and meeting the renowned scientists. And let me have the privilege to introduce Dr. A. Srinivas Rao, sir. He received his BSc, MSc, and doctorate degrees from Andhra University, Visakhapatnam. He started his teaching career in the PG department of botany, VSM College, Ramchandrapuram. Later in 2010, he joined as a lecturer in PR Government College, Autonomous, Kakinada. Later, he did his job at Government Degree College, Mandapeta. Presently, he is working in Government College, Autonomous, Rajahmundry. He is also the in charge of the head of the Department of the Botany. And presently, he has prepared syllabus for 3 market-oriented courses. Number 1, horticulture. 2, seed technology. Number 3, organic farming, assigned by Andhra Pradesh State Council of Higher Education. He is also the author for UG botany textbooks of 1 and 2 semesters. And he is also the member of Board of Studies of Adikavi Nannaya University, Rajahmundry, for botany, horticulture, and forestry courses. He is also the evaluator for National Children's Science Congress activities and DST Inspire programs at district and state level. He is also Mana TV live presenter and also delivered many talks on air. He is also the member in East Godavari District Biodiversity Management Committee and his area of researchers. He is the recipient of JRF, DBT, Government of India, and SRF, CSR, Government of India. He obtained his doctorate degree in the domain area of marine algae, developed a protocol for thallus induction and thallus regeneration. His research interests are marine algae, taxonomy, ecology, psychology, and cultivation and biotechnology. He published 12 research distinguished papers in the journals. He is also the life member of Seaweed Research and Utilization Association, Tamil Nadu. He is the mentor for DST major projects, completed consultancy project sanctioned by AP Medicinal Aromatic Plant Board and AP State Biodiversity Board. Adikavi Nannaya University, Rajamahendravaram, conferred research guideship while working at PR Government College, Autonomous, Kakinada. He is also the educated PhD thesis of Mr. S. Palanivel on freshwater algal diversity of Tamil Nadu, India, submitted to University of Madras, Chennai in 2019. He is also the adjudicated PhD thesis of Ms. M.A. Akhila on efficacy of raw and spent seaweeds as feed ingredients for tilapia culture. Submitted the thesis to Annamalai University, Tamil Nadu in 2019. Further, his name is included in the panel of educators for PhD by Algappa University, Karaikudi and Bharati Darshan University, Tiruchirappalli and MS University, Kanyakumari. He is also the one of the 5 editors of SRU, International Journal of Volume 38. He also has done many seminars and he participated in many events. He organized a national symposium on algae for human welfare, state level seminar on mangrove ecosystem of Andhra Pradesh, national workshop on medicinal botany, national conference on recent trends in plant sciences. He delivered invited talks on 3-day international conferences on advances in algae biotechnology at VIT University, Vellore, Tamil Nadu. He also delivered lecture on 3-day national research meet, developing knowledge management system for coastal biodiversity conservation organized by UNDP GEF IGRI Foundation, Kakinada. He also delivered lecture in 3 days international conference on marine agriculture and pharma biotechnology at JNTU, Kakinada. He participated in many training workshops. He participated in 30 conferences, national and international state level conference. Now I request Dr. A. Srinivasarao Garu, who is also a great privilege to have him at PR Government College. We together worked in that college. And now it is a great pleasure, sir, to invite you to this great day. Thank you very much for giving me this opportunity. Hearty welcome to this workshop, sir. Thank you, Srinivas, sir. He is going to deliver his topic on awareness of fungal diseases. Thank you very much. Over to...
Good evening, one and all. Am I audible?
Yes, sir. You are audible.
Yeah. I'm very thankful to Padmaja madam for a nice introduction. So, I'm very grateful to Dr. Shanai sir and Dr. Sharma sir for giving elaborate talks on the recent researches in fungi area. So my talk is rather very preliminary talk, and my target group is the budding botanists pursuing botany at UG and PG level. So I would like to share my PPT. Is it visible?
No, sir.
No, sir.
Is it visible now?
No, sir. You're not sharing your presentations.
Okay. Just wait.
So can I help you, sir?
Sir.
So go to your Google Meet window, the tab on the browser.
Okay.
And then there would be fifth button present now.
Yeah, that's same thing I'm using, sir.
Yeah, then you go to entire screen.
Okay.
Then there is an image of that window. Now so click on that image. Then you select share.
Full screen.
Entire screen. Then there is an image, just below that option. Small image of entire screen.
Okay. So I have some problem. Just I will share.
Sir, is it allowed for sharing, sir? That fifth button for you. Srinivas, sir? Is it allowed for sharing?
No, ma'am. It is not.
I think it's not visible, Sarah madam.
No, ma'am.
Shahjadi madam? Hello?
Yes, ma'am. Please tell me.
Once you please add that, that thing should be shared with that person.
So you try now, sir.
Sir, once you please check.
I hope now it's visible, sir.
Yes.
So is it visible?
Yes, sir. It's visible.
Yeah,
it's visible, sir.
Slides are not shown here. Your presentation is not visible, sir. Once you please.
Otherwise, I will do one.
Sir, kindly share your window or
I will
tab.
share the. Yeah.
Share window or share tab, sir. Check out.
Just.
Sarah madam. Madam, otherwise tell him to share the PPT to you or Begum madam. Okay. Shahzadi madam. So that you or herself can share the screen. It's okay. He can send the PPT.
Hello.
Hello.
Sir, please send your PPT to me, sir.
Srinivas Rao garu,
Ma'am,
మీ PPT కొంచెం పంపించండి
I have
సార్.
sent the
Shahzadi గారికి
PPT
Sarah
to
గారికి.
your WhatsApp, ma'am. Sarah, ma'am.
Okay.
Sarah, ma'am.
Send.
Sarah, ma'am.
Okay, sir.
I have sent the PPT to your.
Okay, sir.
It's okay, sir. Thank you, sir.
Master. Sir.
Srinivas sir, you please stop your presentation. Then we will present the screen.
Is it okay, ma'am? Now? Have you received?
I have received your PPT, but you stop your presentation, sir. Then we will present.
Okay.
Sir, kindly stop your presentation, sharing your window.
Okay. Yeah. Yeah. Okay.
Okay, thank you, sir.
Thank you, sir.
Now your presentation is visible, sir. You please continue.
Okay. I deem it as a privilege to address the galaxy of botanists and the young botanists who are pursuing UG and PG courses in botany. My passion about this is actually at the MSc level, my specialization is mycology and plant pathology, so that I am associated with this one-day webinar. So today I will talk about this is like awareness on the plant diseases. The seriousness of the plant diseases are threatening problems posed by the fungi on plants and animals. So the first slide, ma'am. Yeah. So as mentioned by Shania sir, fungi are quite different. As per the Whittaker classification, fungi are excluded from the plants and they kept in a different kingdom, that is fungi. So the fungi are the heterotrophs, but according to one school of thought, the fungi are derived from the algae in the course of evolution. That may be a kind of thing because they are sharing the same kind of the thalloid body and having certain similarities also. Though fungi are the that is eukaryotic, like the plants and animals, they are having different distinguished characteristics when compared to plants and animals. So the fungi are heterotrophs. They may live as the saprophytes or the parasites. Sometimes they may live as the symbionts in the form of mycorrhizae. We know that the mycorrhizae is an invisible network that is connecting different kinds of taxa of plants, and through this network, the higher plants are communicating with each other and having the molecular talks. So far, 1.44 lakhs of species of fungi are reported, and in one aspect, the fungi are resembling the arthropods. In arthropods, the exoskeleton is made up of chitin. In the fungi, the cell wall is made up of chitin. So certainly they have the relationship with both the plants like algae as well as the animals like arthropods. So the chitin material is having so many industrial applications, and the chitosan, which is a product of chitin, can be exploited for many purposes. So fungi are causing so many kind of diseases in plants as well as animals. Long back in BC, the Romans realized the importance of plant diseases, and they used to conduct one kind of festival every year. That festival is called as the Rubigalia festival. So that is to pacify the ill effects of the god Rubigus. He is considered to be the god of rust, who is responsible for causing so many kinds of diseases in plants. Next slide, please. Next slide, please. So the world witnessed so many kinds of the plant diseases due to the fungal pathogens. We know in 1841 to 1851, the Ireland faced one kind of famine that is due to a kind of disease, late blight of potato on a potato plant. In Ireland, the potato plant was the staple food for the people, but this Phytophthora infestans, previously it is considered to be a fungus, but now it is kept in a separate kingdom. It is previously in Oomystis of the fungi. So this Phytophthora infestans caused this late blight of potato. So it is responsible for a demographic change in the Ireland country. So the Ireland population came down to 6.6 million from 8.4 million. So previous to 1841, the population was 8.4 million, but in 1851, the population was 6.6 million. So about 1 million people were died due to the starvation of the food, and the other 1 to 1.5 million people migrated to the American continent after this famine. So here you can able to see the visuals showing the famine that is caused a devastating effect on the Ireland population. So next slide, please. Next slide. Hello. Yeah. So the other famine that is considered to be the Sri Lankan famine. So in Sri Lanka, the most of the economy, previously it was called as the Ceylon. The Sri Lanka used to get its revenue from coffee cultivation. So in 1870, about 1870 to 1890, one fungus belonging to the Mycotina, that is Hemilia vastatrix, attacked the coffee plant. So in early days, the coffee exports from Ceylon were about 100 million pounds, that is about 45 million kilograms in 1875. But that came down to the 2.3 million kilograms in 1889 due to this coffee rust disease. A single pustule of this coffee rust on the leaf produces about 1.5 lakhs of spores. So this is a highly devastating disease on the coffee plant. So after this famine, what happened is the entire Ceylon country changed from the coffee cultivation to tea cultivation. So the coffee cultivation is completely arrested, and they went for the cultivation of the tea plants. So next slide, please. So in Europe, we know that grape wine is a important crop for making wine. So downy mildew disease caused by Plasmopara viticola attacked the grapevine crops in 1870s, and that caused great losses to the grape growers. So that also affected the winemakers in the Central America as well as the US also. So next slide, please. So our country witnessed a famine. During the British rule, we used to have a Bengal province. The Bengal province includes the Bangladesh, West Bengal, part of Orissa, and some parts of the northeastern India. So in that area, the staple food is the one kind of variety of rice. So during 1943, Helminthosporium varaceae belonging to the Ascomycotina caused a disease, leaf disease, on the rice crop. That period is coincided with the Second World War, so that people are not getting the rice from the other countries and other places of the India also. So that so many people, about 2.1 to 3 million people died of starvation. Here you can able to see the photograph during that period taken in Bengal. Here it is almost showing a starved bodies of the people. Next slide, please. So we know that the K.C. Mehta, who is considered to be the father of the plant pathology in India. So the red rust of wheat that attacked almost several countries. So that caused a kind of losses in US and also in India. So the Fuchsinia graminis tritici belonging to the Mycology, it is causing the red rust. It may cause the losses up to 45 to 50% in some cases. If it is the stem rust, means it causes about 100% losses also. So we know that always the diseases of the fungi or diseases of any microbe is dependent on the three factors. One is the pathogen, the other one is the host, and the third one is the environment as per the disease triangle. Or as per the disease pyramid, the time may also pose some kind of effect on the prevalence of a disease or epidemic or the pandemic conditions. So here the in Ganga plains, so always the wheat cultivated in the plateau of Ganga area, Ganga river area. There you will find the prevalence of this red rust of wheat. So this also an important disease with respect to the it is affecting the staple food crop like wheat in many countries. So next slide, please. So we know that ergotism. So ergotism is a kind of disease that can be seen in the case of the animals, sometimes in human beings also, who consume the rye plant, that is the produce of rye, that is attacked by a fungus called as Claviceps purpurea. So this causes convulsions, hallucination, gangrene, and also sometimes the loss of milk in cattle. So this is consisting of so many kinds of alkaloids, and these alkaloids are mainly responsible for causing a kind of problem called as ergotism in humans as well as the cattle. Next slide, please. So there are mycotic infections in animals also. The mycotic infections in animals may be of three kinds basically. So the superficial infections, so that appear on only the surface of the skin, particularly in the epidermal region. The other kind of diseases may be cutaneous diseases that may attack the dermal region, particularly they may attack the nails and the hair also. So the subcutaneous infections that attack the muscular regions of the animals. So the world is witnessing the mycotic infections after the COVID period. So we know certain prevalent mycotic infections after the COVID. Let us see very few mycotic infections of human beings. Next slide, please. So candidiasis. So this is generally seen in the case of this is the candidiasis is caused by different species of candida. So candida albicans can be seen in human beings in many organs like mouth, throat, gut, and other regions. So this is normally not causing any kind of severe problems for the human beings. So they are showing certain kinds of symptoms, but not posing any kind of threat for the human life. But recently, in foreign countries, particularly in USA, one species of candida is found to be very prevalent. Candida auris. This Candida auris is considered to be a multi-drug resistant variety and causing many outbreaks in healthcare settings. So that this is posing a threat for the human life in hospitals and other kinds of the health care institutes. So this is one of the recent threat for the humans after the COVID, particularly whoever affected by the COVID, they are prone to this Candida auris very easily. So that this is a kind of becoming a pandemic disease all over the world. Next slide, please. So we know already Shania sir mentioned that mucormycosis, that is this mucormycosis is one kind of threat that is appeared after the COVID-19. So this is considered to be a black fungus by the media. So this black fungus, we know that the Rhizopus species as well as Mucor species, they are mainly causing this mucormycosis. So this mucormycosis, they may affect different kinds of the organs of the patients who affected previously by the COVID, that is the COVID virus. So they may attack the rhinocerebral area, that may cause the sinus spot, and also they attack the brain also. So sometimes they attack the body or the lungs. So they may attack the skin, that is the cutaneous mucormycosis, and sometimes they may attack the gastrointestinal mucormycosis. So this is mainly causing the blood vomitings and other severe problems for the human beings. So next slide, please. So blastomycosis is another problem that is very prevalent during the recent years. This is caused by the species of blastomyces. So people with weakened human immune system, particularly the people affected by the HIV virus and having the organ transplants, and people who are using the corticosteroids, particularly for the tumor inhibiting medicines. Whoever taking this tumor inhibiting medicines, they are very easily prone to this blastomycosis caused by the blastomyces. So next slide, please. So pneumocystis pneumonia, this is another lung problem faced by the people having the weakened immune system, as well as people affected by the COVID virus also. This is caused by the pneumocystis gyrovesi, and the people attacked by this fungus are having chronic lung problem, and the cancer patients also very easily prone to this disease. So people suffering from certain inflammatory diseases, they are also prone to this problem also. And people having the organ transplants or the cell transplants also very easily attacked by this fungal pathogen. So next slide, please. So aspergillosis is another common problem. We know the aspergillus, it is almost a cosmopolitan fungus. This exists in different things, particularly the spores of this fungus are airborne, and whenever we inhale these fungal spores, we are easily prone to different kinds of problems caused by this aspergillosis. So particularly, it is sometimes attacking the bronchial area and pulmonary area. It is also responsible for sinusitis, and it is also causing the aspergilloma, that is a severe lung problem, and pulmonary problems, that is for a long time, that is the chronic pulmonary problems are also seen due to inhaling of this aspergillosis, that is the aspergillus spores. So next slide, please. So cryptococcosis, this is another problem due to two species of cryptococcus, and this causes the infections in lungs, also in the nervous system. So the individuals having the suppressed immune system are more attacked by this fungus species, and they are having the severe lung problems, and sometimes they have the nervous problems also. So coming to the next slide. Okay. Now I stop my talk here. So if any queries are there from the students, I'm very happy to answer.
I request faculty and students, if you have any queries, you please unmute yourself and ask your questions.
Oh, good afternoon, sir.
Good afternoon.
Sir, good afternoon, sir.
Good afternoon. Good afternoon.
Sir, I am Dr. T. Salih, lecturer in botany, sir. One query, sir, small query.
Sir, good afternoon.
Sir, it is any effect of the Puccinia species in human beings, sir?
No, sir. No. Actually, so there is one-to-one specificity always there in the nature. So actually, host and species, host and the pathogen specificity is always there, so that only the plant pathogens are not attacking the human beings, except the toxin. That is the claviceps is producing one kind of toxin, that is alkaloid toxin. So that is affecting the human physiology or the physiology of the animals, that is cattle. So not the fungus. So recently there is one report from the Bengal area. So the plant pathogens are also attacking the human beings. So
So
one
that's why
mycologist.
my asking, sir. That's why recently some of the members, one of the scientists, they are always busy for the search for the fungi. They suffer from the fungal disease, sir.
Yeah. One mycologist from the North India, he working on this medical mycology and the plant mycology also, and he suffered from one disease due to the plant pathogenic fungus.
Plant
I request
fungi, sir.
Sharma
Plant
sir, plant fungus. So I request Shanay sir as well as the Sharma sir to answer this query. Sir, how it is possible? What is the cause for that? Is that due to the environmental changes?
So I don't know
So
much Hello, sir. Hello.
Yeah. How a plant fungus attacks the human being, sir?
Can you hear me?
Yeah, sir.
I mean, I don't know much about that specific case. Okay.
Sir.
But I and my PhD students worked on Coleotrichum.
Sir.
When I was in IMTech Chandigarh, then when we were doing the literature survey, we found that some species of Coleotrichum can cause keratitis. You know that not keratitis, an infection. They will infect the eyes.
Hair.
Eyes, not hair.
Eyebrows.
Eye, inside the eye.
Inside the
eye. Okay.
So see, the mostly Coleotrichums are
as in the case of mucorosis, sir.
Let me explain actually. Yeah.
Sir.
Mostly these are Coleotrichums are plant endophytes, plant pathogens. Yeah, and they are also saprobes. But their association with that human eye or human tissue, I think it's when we did the literature survey and we talked to some of the medical mycologists, we found that that particular person who was infected was a farmer.
Sir.
Okay, and Coleotrichum infection is there in chili and other agricultural crops. So I think probably this particular person got some wound in the eye. Along with the wound, some fungi also infected. That fungus could be Coleotrichum species. Okay. These are like opportunistic, actually, you know.
Yes.
But I don't know much details about that West Bengal case, Kolkata case. But we discussed that issue in our WhatsApp group, but I did not follow it much. Sorry about that. Yeah.
Sir, Coleotrichum is obligate or faculty to parasite, sir?
It's mostly facultative. You see, it can infect.
Facultative, sir.
Yeah, it can infect so many hosts actually. One species of morphospecies of Coleotrichum gliosporidis could infect 450 plant genera.
Okay, sir.
Now the phylogenetically, that species is like 20. There are some 23 phylogenetic species. But traditionally, if you look at the definition, one particular species could associate with 450 plant genera and causing diseases in many of these genera actually. So it should be facultative, right? Unless and until we revise the taxonomy of Coleotrichum. Another thing is Coleotrichum siamens. Okay, it is separated from Coleotrichum gliosporidis. And this
Coleotrichum, is it causes red rot of sugarcane?
Yes, ma'am. Falcatum. Yes.
Only one species, ma'am. Only that is one species, ma'am.
No, I'm
that
talking
is Falcatum species.
No,
Sir is talking about the genus, entire genus.
Yeah. Right. Another thing is Coleotrichum gliosporidis. As I told you, it was reported from almost 900 plant species. So when we did when the taxonomy revised it, one particular species, morphospecies, was divided into almost 30 phylogenetic species. Okay, among them, one is Coleotrichum siamens. And what we thought, Coleotrichum gliosporidis is responsible for many of the diseases of tropical fruits, was found to be not true when we did the phylogenetic analysis and we redefined the taxonomy. So now most of the tropical fruits are actually affected by Coleotrichum siamens actually. That is the name of the fungus which cause the most diseases in tropical fruits. This is how the taxonomy is to revise and improve the understanding of it. Coming back to Coltotrichum species, that particular case, the farmer accidentally got wounded, and during the wounding, I think immunity got probably weak in that particular part. That fungus could actually infect the eye, and that was not definitely not lethal, not deadly. So he was discharged probably in a week.
Sir.
Thank you very much, sir. Thank you very much, sir, for
you.
elaborate explanation about my question, and thank you for Sara madam.
Okay, welcome, sir. Sir, one question from my side.
Yes.
So, as per my knowledge, actually in PG and UG,
Yes.
what I have studied is that generally in immune suppressed human beings or any persons, they are very prone to fungal diseases first. Fungal diseases symptoms
Yes,
can be
madam.
appeared first than bacteria and virus. What is the reason
Yes.
behind it? I want to know, sir. Actually, it's a long-standing doubt in my mind.
Because of immunosuppression itself, it attacks, madam, like alternaria.
Why can't the bacterial diseases and other viral diseases? Is there any dominance in fungi or fungi attack or like that? That I'm asking. So, for example, in AIDS, in HIV infected persons, first visible symptoms are only fungal diseases, not bacteria, not virus.
Yeah.
Why it is so?
Yeah,
So
yeah.
viral diseases are not visible because of viral interference. I know that answer. Why can't bacteria? Why it is so? Why only fungal diseases symptoms can be appeared? That time asking.
As Shana sir mentioned, they may be adapted for different kinds of environment in the early days of evolution itself. Because now Shana sir mentioned that they are existing in so many areas, like highest pressure areas, highest salty areas, like that he is mentioning. So that they may be adapted for so many things. Particularly, most of the fungi are the facultative type of fungi. So very few are like the Ustulago, the species of Ustulago, only the obligate parasites. Very few fungi are obligate, and most of the fungi, particularly the Pythium species and the Fusarium species and other species, they are very adaptive for different kinds of environments. Yes. Maybe that may be the reason. So if at all any kind of tissue damage is there, definitely they can easily attack. So because of their structure also, that fungal thallus, that is the kind of mycelium. That mycelium may be easily spreading. So the bacteria, when you consider the case of bacteria or viruses, particularly if you consider the viruses, they are completely obligate. So in most of the bacteria also, they are obligate. So if you take the plant cases, most of the fungi, the more number of fungi are the plant pathogenic microbes when compared to number of bacteria or number of viruses and other microbes. So about 8,000 species of the fungi are causing plant diseases. Sir, Shanai sir?
I want
you
to
want
add,
to add anything, sir?
Yes,
Sir.
I would. Yeah, because I recently learned about mucormycosis. Yesterday we had many lectures. One of the lectures was on clinical fungi and public awareness of it. So in that lecture, Professor Dr. Vijayalatha Rastogi mentioned why mucormycosis was so rampant during COVID-2. You know, COVID second wave. You know, I mean, mucormycosis
Second wave and third wave also, sir. Second wave and third wave also, sir.
Yes. So I think it could happen in normal days also when COVID is not around. But during COVID days, mucormycosis was so rampant, infecting so many people and leading to death of 5,000 people. Okay, there were many explanations. One of them was that so the oxygen you give, that is not clean and all those things. What Professor Vijayalatha Rastogi madam told yesterday, because of antibiotics and so many medicines were given to these COVID patients, the system became slightly acidic. I don't know what system she is, maybe the blood system or I don't know which, or lung system, I don't know which system, okay. But because of this slight physiological change, which allowed the growth of this mucor and related fungal species. So basically, there has to be some immunity or immune-related changes, or immunity is compromised actually. So that is how the even healthy people get affected when their immunity is weak.
Sir. So they used the steroids also, sir. That suppressed the immune system, sir. So that the fungus attacked the people.
No, but not. Even I was on steroids. I did not get mucormycosis. See,
no, sir. In second wave and third wave, they used more number of steroids when compared to. Actually, the people having physiological disorders also are responsible for this problem, sir.
Basically,
disorders like
looks like there is sampling data. The sample size is not very good, and we are not able to make very concrete conclusion why mucormycosis was so rampant during COVID at times. Actually, one of the explanation was the cleanliness, hygiene issues in the hospitals. Okay, because these mucormycosis are normally present in the soil and others, that other stuff.
Yeah.
But then if the hospital environment is not clean, probably this already the poor immunity people might get affected. But another explanation was given that because of steroids, as you mentioned, and other medicines, antibiotics and other stuff, the system of maybe one of the body systems become acidic, that allowed the rampant growth of these fungi, especially in the nose and all, right? So the black thing started with the nose and all. Afterwards, some people lost their dental system and all. So many bad things happened. Yeah. Thank you so much. This is what I learned recently. I am not an expert in medical mycology.
Sir, one medical mycologist is there. He retired from the Kolkata lab. He is working in. He is still after retirement. He is working in Kakinada as a practitioner. His almost age is 85. So he is curing so many mycological diseases on the skin, sir. Particularly this psoriasis. He is treating psoriasis using one psoriasis, sir. Psoriasis.
Okay, sir. Thank you, sir. I think we will discuss separately. Madam Sarah is waiting for us to stop the discussion.
Thank you.
Okay, sir. Thank you, sir. Thank you.
Lakshmi, madam.
Yes, ma'am. On behalf of all the faculty of our college, we thank the resource persons who are well-recognized figures with high renowned achievements in their respective fields. We are grateful to all of you, sir. I would mention your names too. Thank you, Dr. Belle Damodara Shenoy, sir, Dr. Rohit Sharmaji, and Dr. A. Srinivas Rao for accepting our invitation and being resourceful persons to deliver wonderful, innovative, and enlightening talks today. Thank you so much. Now I would like to draw upon the closure of the particular webinar, and I would now like to call upon Dr. P. Sarah Madam to give the closing remarks of this particular webinar and enlighten all of us as to how the particular resource persons have provided the participants with great knowledge enrichment. Over to you, Sarah Madam.
Thank you, madam. I'm thankful to Commissioner of Collegiate Education, Dr. Pola Bhaskar Garu, Joint Director R. David Kumaraswamy Garu, Regional Joint Director Dr. C. Krishna Garu, Principal Dr. K.C. Satyalata Madam Garu, for giving the opportunity for conducting this national webinar on recent concepts and perspectives in mycology. On behalf of organization, I'd like to extend my heartfelt gratitude to all resource persons, Dr. Damodar Shenoy sir, Dr. Rohit Sharma sir, Dr. A. Srinivas sir, for their informative sessions and immediate response in correspondence. Thanks to all the participants who made this webinar a memorable one in the field of mycology, and wish all the participants for better future. At last, I would like to extend my heartfelt thanks to Vice Principal Y. Padmaja Garu, Organizing Secretary Dr. N. Srinivas Garu, and the entire technical team, especially Shahjati Madam Garu and academic coordinator and today's anchor, Dr. N. Lakshmi Garu, who made this webinar as a successful event. Thank you for giving me this opportunity. Thank you.
Thank you, Sarah Madam, for your wonderful closing remarks. It was really a great webinar, which was organized by you with great efforts, strenuous efforts, in fact, for the last few days by you and Dr. N. Srinivas Garu. Now I would like to call upon Dr. N. Srinivas sir, the in charge of the Department of Zoology and the IQSC coordinator of our college, to give, or you can say to present, the vote of thanks on behalf of the entire college and the team. Over to you, sir.
Good evening. Very good evening to one and all. Hope I am audible. Am I audible, madam?
Yes, sir. Yes.
Yes, sir.
Okay. At the outset, I am very much thankful to all the participants and resource persons for staying throughout the webinar sessions. So, right from the beginning onwards till now, all the participants are there. On behalf of all the organizing team, we thank all the participants. I take this opportunity to propose a formal vote of thanks as organizing secretary of this webinar. First, we, the Government Degree College Ramachandrapuram family, express our deep sense of gratitude and thanks to our Honorable Commissioner of College Education, Dr. Pola Bhaskar Garu, for his encouragement, support, and vision. And I take this opportunity or this platform to express our heartfelt congratulations to our Honorable Commissioner Garu for his great achievement in achieving Scotch Award recently. We also express our sincere gratitude and thanks to the members of the Honorable Commissioner of College Education, that is JD Sir, AGO Madam Garu, and OSDs for their wholehearted support to our college. Our sincere thanks goes to our Regional Joint Director of College Education, a true academician and a well supporter of our college, and who is the guiding force and guiding us in various aspects and various points for successful completion of this webinar. Similarly, I thank our Honorable Principal Madam Garu, who is the dynamic principal. Under her stewardship, this webinar has made a grand success on this day of Gandhi Jayanti and Lal Bahadur Shastri's birthday. Our sincere thanks goes to the organizing team and the advisors from our college, as well as the advisors from other parts of the college, other parts of the Pan over India. There are advisors from other institutions over India. Next, our sincere thanks to our students, members who have participated, and sincere thanks goes to the technical support team, and our sincere thanks to the press and electronic media for covering this webinar. Thank you, one and all.
Thank you, tell you one thing
for
to all
such a
the
nice
participants.
vote of thanks. Madam.
Sorry for interruption, madam. Just one minute. So all of you, please be with us in the WhatsApp group. We will send you the feedback link to your respective mails so that you will get a certificate within couple of days. So sorry for inconvenience. Actually, we are preparing the certificate. Thank you. Madam, you can continue.
The feedback link will be sent to the mails of the respective participants. I request all the participants to check your emails and try to fill the feedback form by clicking on the link. and then certificates will be issued in a few days. Kindly bear with us. I hope all the participants would agree to that. Thank you so much, all the participants. And now, last but not the least, for the closure, we are going to have our national anthem. And I request all the participants to join hands together and respect our national anthem and salute the nation with due respect. Thank you.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all for your kind participation. We can call it a day and we can close the seminar. Thank you all of you once again from Government Degree College, Ramachandrapuram, signing off for today.
From all over the
Thank
world,
you all of
mixing first-person shooting, hypernatural powers, and hundreds of tactical strategies.
మధ్య ఎక్కడ ఎక్కడ